Good morning, everyone. Thank you for joining. Good morning or good afternoon, depending on what part of the world you are from. We're really excited for get started on the second day of our NCPI workshop. And we've got an exciting agenda planned for today. Another filled agenda with a lot of great ideas and information coming in. We'll break today's agenda up into a few sessions, including some interoperability driven science. We'll hear about some NCPI working group updates. We'll have a third session of technical aspects of interoperability before jumping into some concurrent breakout sessions. After that, we will wrap up the workshop for the second day and conclude our workshop. And we're really excited to hear some of the ideas today. So why don't we, at this point, hand off to Jonathan Tedds from ELIXIR, who will present to us some ideas about the ELIXIR Cloud for European Life Science. I will stop sharing at this point, and Jonathan Tedds, if you are ready, you can go ahead and kick us off.
So, yeah, very pleased to be here and able to present to you to this meeting on the ELIXIR Cloud and our work across mostly European-based member states to present this work to you. Apologies, slight delay on the load here. Okay, so hopefully you can see that okay. So I'm Jonathan Tedds. I'm based in the Cambridge hub of ELIXIR. And I'll try and explain a little bit more about what ELIXIR is by way of introduction, and then just an overview, obviously, in the time we have, of the kind of things we're working on in the compute platform in particular towards building the ELIXIR Cloud. And then I'll come on to a sort of a specific example in one of our nodes. What do I mean by node? I'm looking here at a map of Europe. You can see that we have this large number, where are we now? 22 formal member states. So it's an intergovernmental organization. We're sort of administered through the European Molecular Biology Lab, but we are a separate legal entity that follows the same model. And this means it gives us independence from, for example, some of the best known structures and avoids some of the more difficult political issues that you might have around EU membership or not, for example. So we have the full member states, a couple of countries, one in particular in observer status at the moment in Cyprus, and then we have sort of strategic partnerships. I'll come on to this with Global Alliance for Genomics and Health, and we work with a lot of international organizations in multiple global projects. Australian Biocommons, for example, I mentioned them as well as a good collaborator we work with. So what do we cover? The nodes or member states, they are collections of a number of institutions in each of the countries. And obviously the focus is on life sciences, bioinformatics, but not just human data, also covering a wide range of communities, as you can see. So we have communities of practice around, for example, the Galaxy tools ecosystem. But then we have specialisms in plant sciences, metabolomics, and so on and so forth. So you'll see there's something like 13 formal communities currently, and they have to they produce white papers, and they then are approved or not within the ELIXIR domain if they're working on particular problems at that moment in time. More bottom up is we have currently 12 focus groups, which in some cases may go on to become communities within our structure, or they're just working on a particular short topic that might only be a few months duration. But knitting all of these together is our 5 technical platforms. So compute, training, interoperability, data, and tools. And you'll see the links to a bit more detail on those. And we bring these to bear. We bring all of this joint work to bear in our communities and in a range of, in particular, EU-funded projects, but also a range of other projects too. So we're not a huge funder ourselves. the funds that we spend are just for specific implementation studies, and they're based on sort of membership fees from each of the member states. But most of the resource we work with, so to speak, is through various EU projects that we either participate in, lead, or in some cases coordinate. And there are many hundreds of services. Here's perhaps one of the most widely used at this point across ELIXIR, which is what used to be called our ELIXIR AAI service, but is now transitioned successfully over the last few months to life science login. So ELIXIR has been building upon the Common Internet Standards and OpenID Connect and so on over a number of years, working through Corbel and ARC projects in Europe in particular, but working with many other research infrastructures, and basically building this identity management and access management service. So the idea is that a typical user base or a typical researcher in a university, but could be elsewhere, uses their home credentials, associates that with a life science login, and can then access multiple services across multiple infrastructures. And there's a few example life science ones shown there. So ELIXIR was the first to migrate all of its users to this new service, which was built through the EOS Life project, a large sort of life science research infrastructures project in Europe. And we now have many thousands of users successfully migrated to this life science login service, which, and gradually, one by one, the other European life science infrastructures will come on board. They've been working directly with ELIXIR all the way through the project to make this and enable this to happen. And you can imagine, as you can imagine, this fits into a sort of larger European open science cloud picture for how we want to address identity access and management. The services that use life science login will gradually be migrated over the next 12 months, but they still work via the previous ELIXIR service silently in the background. So there's no blockage on using services for life science login users. Okay. So whoops, gone forward too many. So to start to get a bit more cloud specific, the kinds of services and solutions that we work with across ELIXIR, just to be clear, these are driven and built by our nodes, so our member states, leading organizations. ELIXIR doesn't operate the services directly from the hub in most cases. They're nearly all operated by the nodes. ELIXIR acts as the coordination point for all of this work, and therefore we're in a good position to push common standards, including through cloud. So typical examples in terms of maturity, you've obviously got a very mature, in many respects, very mature community around the Galaxy project. There's a whole European entity for that, who, as many of you will know, have web-based platform for reproducible computational analysis. A much newer thing, so less mature as a service, is what's called Workflow Hub, which is this registry we've been building at a European scale for describing, sharing, publishing computational workflows. And then at the more sort of proof of concept stage is the kind of work we do through the Global Alliance for Genomics and Health Driver project, which ELIXIR leads in cloud and AI. So we are one of the driver projects that contributes towards the standards that are developed through the cloud workstream, the researcher identity workstreams, and so on. And so I'll say a little bit more about that in a moment. The way we go about this work is to represent ELIXIR stakeholders, obviously in Global Alliance, and promote those standards across ELIXIR. We're working on prototyping real-world use cases in, for example, large human data projects by developing these proofs of concept and deployment at ELIXIR nodes in their compute centers. So we obviously consult on integrating these standards and provide technical support critically for And I think many of you will know, but just in case not, these are the key standards that we've been working with through this ELIXIR Cloud and AI driver project. So Tools Registry Service, Data Repository Service, the WES Workflow Execution Service, and Task Execution Service. And one of our nodes, EBI, European Bioinformatics Institute, has led or co-led the development of the version 1 of that task execution service. And Passport, which is this wider passport researcher identity standard, is now a standard in Global Alliance, and that's sort of been co-led by ELIXIR and Google, or at least the first 2 implementations of that passport standard were done by those 2 actors. And then this is actually a relatively old slide now, but just to illustrate how we've worked alongside international collaborators through, first of all, this is a term we used, a moonshot demonstrator we performed, where we implemented all 4 of those cloud standards across 6 different ELIXIR nodes, so in 6 different countries. And this was just a demonstrator using 1,000 genomes non-sensitive data as a proof of concept technically, but you can see those other actors were also participants in that. There's since been much more work through the Federated Analysis Systems projects to take this to further and to start to work towards real-world implementations, and in particular to integrate the passport standard with those cloud standards. And so an example of the sort of reach and how we were able to use our network potential in ELIXIR was obviously during the COVID response, where you can just see a long list here of different compute facilities across Europe and how they were deployed in particular ways for particular assistance to researchers reacting quickly, and obviously international collaborations worldwide to work on particular aspects of the pandemic response. So I wanted to close by just giving a more specific example from one of our nodes, one of our largest nodes, of course, which is the German node, and the Denby network. So you'll see a large number of projects, a federated cloud compute system with a number of individual compute centers within that. And they work very closely through the ELIXIR Germany node with partners, and they co-lead some of the tasks in the compute platform, for example. And of course, the European Centre for Galaxy, the large galaxy community in bioinformatics, is based in Freiburg. So it's a fully academic cloud federation, first established 2016, free of charge for academic use inside the German state, and maintained, as I say, through these 6 cloud centers. These will no doubt be out of date already by now, but just to give you an idea of the range of scientific computing available for researchers in Germany, and then to give you an idea of the sort of increasingly large number of projects making use of those facilities. And the important point to note here is how, as far as possible, they reuse the standards that we're co-developing and coordinating through ELIXIR and its projects, but obviously working internationally as well. But you get an idea here of the growth of projects. There's a sort of OpenStack flavor of what they can offer to researchers, and then a more bespoke VM-based, simple VM-based service for their users. And they were also quite early, so more than 2 years ago, they integrated the AAI service so that researchers can bring together multiple services using that resource. And then finally, just an example at the more techie end of things of how we're working through that Global Alliance driver projects to work on specific workflow execution service implementation. And the kind of work so developing a WES for SnakeMake and Nextflow, So this is in Germany at Charité University in Berlin with a couple of collaborators there. But although that implementation and that project is funded nationally within Germany, they work closely. There's a Slack board for all the people working on this across ELIXIR Cloud Network to help and provide experience and so on. So my last slide is just to illustrate, perhaps as talking points, now or later, I realize there's not too much time, a kind of gap analysis from the experience to date. And I think they'll be fairly familiar to anyone working in our context. How to resource and pay. They're obviously relatively well-established, simple mechanisms in commercial clouds, but they're not always easy to implement in an academic setting. Then you have the issues of access control, and I've sort of illustrated how we're addressing and seeking to address that. And we're planning for this major new European genomic data infrastructure project, which will start this autumn and which ELIXIR has been, at least so far, coordinating. We'll see what the final version looks like in the autumn. And then as far as sensitive data is concerned, how to secure data beyond access control itself, how to integrate standards such as using standards such as CRIP for GH, how to integrate that with the cloud APIs. And I think the biggest learning lesson of all from the pandemic response was the probably well-known gap in each individual group, but sometimes we don't talk about it enough. We could provide lots of compute facilities. Lots of research groups working on the pandemic response needed those compute facilities. Huge lack of the kind of people who can actually help them on board to use the relevant service. And this is often a gap, of course. So this is something that's a really high priority for us to help to address that. Okay, I'll stop for now.
Thank you, Jonathan, for that wonderful talk. Does anyone have a question they'd like to raise with Jonathan? I see in the chat there's a question. Do you have any specific plans to include multi-party homomorphic encryption, MHE?
Oops. I think I'm still unmuted, aren't I? Yeah, not that I know of. So I'm familiar with multi-party with MHE. I won't try and say it again. But I don't know of specific examples at the moment to do that, but I can imagine that as soon as I ping people on the ELIXIR Cloud Slack board, that there will be people who tell me that they have been looking at that. So I'm afraid, I don't have, I can follow up with you if you'd be interested, and I can connect you with anyone who's looking at that specifically. You make a note, so that's Heidi, isn't it?
So I have about 1 million questions, but I'll try to keep just to one. You must have had some thoughts and discussion about kind of building your own cloud platforms versus adopting a commercial cloud platform. I'm wondering, what sort of led you in one direction versus the other?
So I think mostly this is driven by the nature of our entity, if you like. So ELIXIR is an intergovernmental organization. What we principally do is coordinate these services from our nodes. And so what that means is that if someone comes to us and says, we need some compute resource to do X, in the case of compute resources, we have to point them to a range of nodes who offer those, for example, if they need high performance computing, we can point them towards the relevant nodes that do this, and then they can negotiate for that access. So in other words, ELIXIR itself doesn't provide those compute services. In practice, each one of our nodes is, of course, working with the commercial as well as their own infrastructure to varying degrees. So this is a constantly evolving picture. And we have a hybrid cloud task, which is looking specifically at this in ELIXIR and trying to learn lessons across Europe from the experience so far. So, so depending on which node services you use, the European Bioinformatics Institute has now after 2 years, I believe, signed agreements with AWS and Google, as well as their own infrastructure for, depending on the use case of the research project in question. So I guess what I've done is describe where we are now, and I think it's still an open question as to what extent we will move more one way or another in future. We're still, effectively, the nodes themselves are still analyzing their experience with this, and that is informing what we do together in future through ELIXIR.
Thanks very much.
I see there's a couple additional questions in the chat, but in the interest of time, I think that we might want to move on. Jonathan, feel free to reach out through the chat or through
Sure, yeah,
or however works for you.
That's great. Yeah, thank you.
If you could
stop sharing.
First of all, yeah, if you could stop sharing, number one, and then number two, thank you very much for this very enlightening talk. We're really excited about some of these things that are going on in ELIXIR, and looking forward to ways that we might interact together and promote the interests of both NCPI and ELIXIR. So
that's great, thank you.
Okay, so at this point we can advance to the talk of Melissa Wilson, who will give us an update on her project Sex Chromosome Complement Aware Alignments.
Great, thank you for sharing, Stephen. And I'll just start off by saying, so this, can we go to the next slide, please? Yeah, so this is really spearheaded by Brendan Pinto in my lab, and I feel like very small fish. I say this at every meeting I go to of the NCPI because we watch these talks about just massive amounts of data, and we are one of the very small fish trying to figure out how to use it, right? So we are one of the use cases that was asked about in the chat, not on the European side, but on the NCPI side. So I'm going to talk about sex chromosome complement aware alignment. Brendan's here also, so he can chime in about some of the challenges we've had, but next slide, please. Just a huge thanks to Brian, Michael, and Samantha, who have helped answer some questions. So we've, do what you do best as a bioinformatician, which is search the internet for answers to your questions. And then when we come up blank, ask people for their deep insights to these. And I have to say, just we've gotten a lot of guidance from all three of these people. And so that's been really helpful. Next, please. In particular, so Brendan is a really excellent genomics researcher, came to my lab from studying reptile sex chromosomes, and my background is in evolutionary genetics and genomics, some in humans, some in non-humans, but really we focus on sex chromosomes, both identification, looking at genetic variants, looking at genetic diversity, and then we've kind of found ourselves in this area of doing some small software. I would say it's really much more intellectual contribution. We're not software designers, and we can get into that, but writing software to fix our problems or the problems that we see with calling variants on the sex chromosomes. We can get into this. We've published a few papers lately about how some of the default pipelines just don't accurately account for X and Y chromosomes, and that then leads to false positives, false negatives. And because we, in our little corner of the world, care a lot about incorporating X and Y, then we've been working on trying to improve methods here. So thanks to Brian and everyone at NCPI for helping write the application and getting it in to get some small pilot funds. Next slide, please. We are analyzing all the genomes here, and it's just a little mind-boggling to think about how many genomes are there that we can work on. And so I'm going to tell you about our grand plans to analyze all the genomes. Spoiler, we didn't get there yet, but next slide, please. The root of the problem that we're interested in affects X and Y chromosome variants called in every genome project ever. and in next generation sequencing data or second gen, any of them actually, because the X and the Y chromosome in humans and mammals share sequence similarity due to their shared evolutionary history. So they were once a pair of homologous autosomes. They're now homologous sex chromosomes. And although they've diverged from each other, so the Y chromosome has lost about 90% of the gene content it used to share with the X, they still share some regions, these little blue ones in our cartoon, that are 100% homologous. They do have some SNPs between them, but they're 100% homologous, just like autosomes. And then the regions in the green share some sequence homology. And so next, please. When we think about normal genome alignment, we first think about the human karyotype. So we're doing biology informed alignment. And typically a sample from a human will have 22 pairs of autosomes, one from genetic mom, one from genetic dad, and either 2 X chromosomes or an X and a Y. But when we do our typical alignment, next please, we use a single representative of each autosome for our reference-based alignment. I know there's some methods that are working on incorporating alternative haplotypes, but in general, we incorporate one copy of each of those, but the X and the Y, we incorporate both of them, which means you have 2 regions that are 100% identical in our reference genome that we use. And originally, we had developed an approach to try to correct for this. In XX samples, we should hard mask the Y chromosome completely. Oh, next, please. Sorry. Forgot I had a slide about it. Right, in XX samples, we'll hard mask the Y chromosome. So we force any of the reads that are similar between X and Y in a sample that doesn't have a Y chromosome, right? They shouldn't be aligning to the Y, but they do because of shared sequence similarity. So hard mask the Y and force those reads to go somewhere else. Usually they go to the X. And in XY samples, at a minimum, we can hard mask those pseudo-autosomal regions that are 100% or very similar in sequence. And so we showed this with a small number of samples in GRCH38. Now there's a new telomere to telomere Chem13 reference genome. And what we want to see is, we see that it's a problem on a small scale. How much of a problem is this in terms of missing variants, because we have lower than expected sequence coverage on the X in a standard alignment, because some of those reads are going elsewhere. They're going to the Y chromosome. So next slide, please. So we have our little workflow over here, which is that in what's available. So we requested access to GTEx, all of the Genotype by Tissue Expression Consortium, and TCGA. I'm happy to talk about why it was much more challenging to think about accessing kids first and top med. So a couple years ago, we were thinking, great, we'll just run through TCGA, GTEx, which I've used before, kids first and top med. The brief aside is that when you request access, you can request access through dbGaP to all of GTEx, to all of TCGA, but not, in my experience, to all of top med or to all of kids first. And it's been much more difficult in my experience to identify which samples have DNA and RNA, which is what we wanted in the long term here, in some of these data sets versus others. So from a user perspective, it's not quite as clear even to figure out what data is there to know what to request for the project we want to do going forward. What we're going to show here is analysis of 15 XX samples from GTEx consortium, because what we learned from the last NCPI meeting is that when you're running in the cloud, what you should do is 1, get it working locally on a sample. 2, get it working on one sample on the cloud. 3, do a small set on the cloud, and then expand out to all of your samples so that you don't eat up all of your compute credits with simple errors that you could solve on a small scale. And so the workflow here, which is just kind of in the background here for everybody, right, is we get CRAM files, so we have to convert the CRAM to BAM, strip the reads, because our problem is the reference genome, and then get our FASTQ files and then remap to the Chem13 reference using our sex chromosome complement aware reference genome, then call genotypes, then call individually call haplotypes of GHK, and then call variants. It's pretty straightforward. And Brendan's here, but I want to say that that process also of going from working locally to working on the cloud had many more challenges than we thought it would. But let me show you that we actually did get some data, which is all Brendan, and really wonderful. Oh, sorry, I keep clicking my slides here. Wait, next, please. So we have some super cool data. So what we're going to show is the difference between SNPs called with a standard reference genome, and then what we're going to call unaware of sex chromosome complement, and then with XY aligned, so aware of the sex chromosome complement. So how many straight up SNPs are called? So, okay, yes, I have reached my max limit. So we're going to have some visitors here, and that's just how it's going to work. So what we see though is that for the X chromosome, we dramatically increase the number of SNPs that we call here. For the chromosome 8, there's a slight decrease, but I wouldn't say that it's significant. But for the X chromosome, we do see a significant increase. We can't confirm that all of them are true positives, but from some simulation data that we've done in the lab, we don't see in the simulated data that we generally increase false positives. We almost always reduce false positives and increase true positives. So even if some of these are false positives, the bulk of them are true positives, increase in the total number of variants called across those 15 individuals. So we're getting a 15% increase across the X chromosome as a whole. But like I mentioned, there are multiple regions of the X at the beginning. Next slide, please. So if we break down the X chromosome into those regions, we have the pseudoautosomal region, which is about little less than 3 megabases. We have the X transposed region, about a little less than 5 megabases. And then the rest of the X chromosome, which we call non-par X, about 150 megabases. And so we see the biggest improvement here is in the pseudoautosomal regions, which have the highest sequence similarity. A lot of increase in the X transposed region, and then still quite a bit of improvement in the non-par region. So we're seeing that this sex chromosome complement aware approach, when applied broadly, at least across more GTEC samples, is improving or increasing the total number of variants called. And based on the simulation data, we think it's improvement. Next, please. And we did one other thing, so filtered out to just the coding regions. So this is exon sequence. And so we wanted to see how much is our improvement if we just restrict to coding sequences. And it's very similar. So it's actually slightly higher, which is kind of cool to me, and would be expected because we expect sequence similarity to be more conserved in coding regions than in non-coding regions. And so we're seeing pretty good improvement. Sorry, keep catching myself. Pretty good increase in variants called across the X chromosome. So yeah, we should be doing this, right? And next slide, please. We didn't analyze all the genomes. We analyzed some of the genomes, and we'd like to analyze all the genomes, but over the last 8 weeks or so, which is just run into many challenges with implementing in Terra. And so I'll go to the next slide, please. So Brendan very helpfully clustered these into 2 categories. So the first was like I mentioned, in moving from the local environment to in Woodle. So my lab generally uses SnakeMake, and so we're moving from SnakeMake to Woodle, and that's fine, right? That's going to take some time, but Brendan got that up and running pretty quickly locally. It's a different workflow manager, and we knew that would take some time. But the challenge was moving from the local environment to porting it over to Terra. And some of these workflow errors that were cropping up, the error messages weren't entirely helpful. We didn't really know where to go to look for problems because they worked locally. And so trying to figure out the test case, what you do is like, okay, pull it down and see how you can get it to work locally, but it already worked locally. And so running into some errors moving up, not even scaling up, just moving up to the cloud with error messages that were non-specific. Next slide, please. And then the next one had to do with something what Titus was talking about. It was just the data localization during analysis. So trying to figure out where the data was, making sure that GATK and Terra and our code and everything understood how to describe where the data is and how to access it. And there's some workarounds. So copy everything into your working directory, call it as a string. So it's not that there aren't ways to work around it. It's just that it is a frustrating challenge. And I can imagine for labs that are not used to doing bioinformatics, period, and then trying to move to the cloud, that this could stall or completely stop people. So next slide, please. And maybe, again, small fish here. So what was frustrating for us especially is that these errors are not new and appear to have been known, right? So we do the Google search, find out that some of these errors are known and that there are some workarounds for them, but the workarounds aren't made immediately apparent when you're first starting out. So it seems inefficient to spend time to troubleshoot, to figure out that the error has been known for months or years and then have to solve it again. I think that could hinder uptake of this, right? Like in the grand scheme of things, once we finish everything's running now on 15 samples, we can scale up now to 10,000 GTEC samples. That shouldn't be a problem, but it's taken us 2 months to get to this point. And when we write the paper, it will say, we ran it on the cloud and it took an impossibly short amount of time to run through 10,000 samples, right? Like that's super exciting. And when I watch all these talks, you can rerun all of TCGA in a week. Like, what? That's amazing. And it doesn't emphasize as much that you're gonna hit your head against the wall for 2 months while you're trying to figure out how to get it to run in a week, which is fine. That's bioinformatics. I know that. Next slide. So our summary is that biologically, we can do some really cool things with improving or increasing the variance called on the sex chromosomes. And now I'm confident that we can do this at scale on Terra, but it's still going to take us a little while longer to figure out how to do this. And then our next step, which is incorporating RNA-seq. So it could be really useful for adding some Terra-specific quirks. I don't know what all of the best options would be, but thinking about what are some common challenges that people run into that if they can't be solved on the dev side, how could we give people instructions so that when they run into these errors, they have quicker solutions to solve it. So that's my last slide.
Thank you, Melissa, for that nice talk. Are there any questions for Melissa at this time? I think we might have time for one.
Hey, Melissa, this is Tiffany. Just a comment. I lead the user education and frontline support team for Terra, and I really appreciate you sharing this feedback with us. And I'm definitely going to be taking a look at what you've run into and trying to see if there's ways we can, either, like you said, improve documentation, if there's ways in which we can improve getting focus around some of the the development or engineering priorities that you mentioned, the things not getting fixed. I'm happy to follow up with that. So just wanted to say thanks for sharing that.
Yeah, no, I mean, it's a point of pilot data, right? Is to figure out what you're piloting. And I'm super excited that we're gonna be able to do this. not sure that Brendan is as excited. I think I partially recruited him saying we're going to analyze. I think I said we're going to analyze all the things, right? We're going to analyze all the things, and it's going to be super exciting. And so I just want to, I guess our purpose of being a little fish in this test case is to say like how might other little fishes respond. Yeah.
Yeah, and this is Adam from Kids First. Again, I just wanted to thank you for being an awesome use case, and really your points about sort of the mental model of accessing a data set, like accessing all kids first versus accessing all TCGA, highlights some of the potential need for us as a community to do a better job communicating about differences between studies in ways that Kids First is comprised of many studies that are independent of each other versus a single study, even though it's under one umbrella, and what that implies for users. So it really highlights some requirements on our end to do a better job communicating on the differences between programs that are sort of out of our hands, really, or just out of the context of consent-based projects and their sourcing. But I really appreciated your framing and sharing your challenges and successes.
Oh, Adam, thanks. And I do want to follow up with you because we have some, forever ago and in the before times, I remember talking with you about these sex differences in pediatric cases that we'd love to get at. And it's,
it's definitely well known in cancer in ways that really we'd love to follow up on, and likewise in structural growth defects. So let's follow up and support your access and implementation in ways that unfortunately will not be uniform and say all of Kids First, but still we can support that process, and it still can be fairly rapid through the DB-GAP framework.
Oh, yeah, no, and I think it can be. I think I just came in, I came in growing up as a bioinformatician in TCGA and DB-GAP, right? And
exactly.
So
Totally agree. Yep.
I think I would have been like, what? Look what you can do with just getting one. But I would have known those different use cases. And so I appreciate especially the informed consent component of it, right? Because not all of those projects are gonna say like, yeah, you can analyze it just because you want to look at sex chromosomes and add those, right? Like they didn't do it that way. And so
But it's important to know to recognize some of those barriers to entry. So I really appreciate that, and let's definitely follow up because your results are really exciting and can be clinically significant in the frameworks of the pediatric and developmental context.
Wonderful. Thank you for all the questions and comments. Rebecca, I see that Becky, I see you've got your hand raised, but in the interest of time, I think we'll need to move forward and continue on with our next talk, which will be given by Nara Sobreira from Johns Hopkins University. And her talk is titled Genome-wide Sequencing Analysis to Identify the Genes Responsible for Enchondromatosis and Related Malignant Tumors.
And good morning, everybody. Sorry, this is the COVID voice. But no, I'm fine. It's just a little, if I cough a little bit, you know where it's coming from. So I'm going to present another small fish project. And also most of the work that is presented here was done by Rehm Martin in our lab. So the idea that we are trying the question that we're trying to answer here is we're trying to identify if pathogenic variants related to the HIF-1 alpha pathway are mutated, if these genes are mutated in patients with Olea disease and Maffucci syndrome, and if these genes are mutated on patients with gliomas and chondrosarcomas. So why gliomas and chondrosarcomas? Because as you see here in this graph, 30% of the patients with Olea disease or Maffucci syndrome have chondrosarcomas, and about 10% of the patients with Oller and 4% of the patients with Maffucci have gliomas. So we asked if the same genes that we were finding mutated among the patients with Oller and Maffucci would also be mutated on individuals with isolated gliomas and chondrosarcomas. We have found that 7 genes on the HIF1 pathway are mutated in about 25% of the patients with Oller disease and Maffucci syndrome that we have analyzed so far. We have analyzed about 100 individuals so far. And these are rare diseases, so this is a large number for the disease that we are looking at. And these numbers got us very interested on this pathway and on these genes. So we asked what are the data sets that are out there for patients, for individuals with gliomas and chondrosarcomas that we could look at. And our idea is to get the samples that are coming from patients with gliomas and patients with chondrosarcomas and look at the variants that we find that are germline for these groups and the variants that we find that are specific tumor variants. And among these two sets, check to see what are the HIF1 alpha regulated genes that are mutated in these sets, in these groups of tumors. So we got together a list of 836 genes that are HIF1 alpha regulated genes that we are interested on to look germline and somatic in the patients with gliomas and the patients with chondrosarcomas. And then we also want to do a burden analysis for each one of these sets of variants in these patients, and then compare the variants that we are finding among the genes that are HIF1 alpha regulated and the variants that we are finding that are in the genes that are positive for the burden analysis. So to do that analysis, we went to check what are the data sets that would be available for us to investigate beyond our patients that we had sequenced with Oller disease and Maffucci syndrome. And we found few sets of sequencing that we would like to analyze. So we found the pediatric brain tumor atlas data set, the CBTN and the PNOC. And we found also with brain tumors, and we found also a group of sarcomas patients in the PNOC that we would like to access. And that's on top of bringing together our own samples. And our own samples have been sequenced as part of 2 projects. So we have 61 trios that were sequenced with whole genome sequencing as part of the Gabriela Miller Kids First Pediatric Research Program. And we have 33 other probands that were sequenced with whole exome sequencing that were sequenced as part of the Baylor Hopkins Center for Mendelian Genomics and that are stored in NView. So basically we had our own set, data is stored in Cavarica as part of the Kids First, data is stored in NView as part of the Baylor Hopkins Center for Mendelian Genomics project. And then we had these extra samples that we would like to analyze, some of them available through the Kids First Data Resource Center in Cavarica, the Pediatric Brain Tumor Atlas, and some of them available through NCI CRDC, that's the chondrosarcoma part of it. So our project was to have access to all these samples in one place where we could go on with the analysis that we were planning to do. So to do that, we started with the Pediatric Brain Tumor Atlas data sets, and there were 998 probands available through the CBTN and 79 probands available through the PNOC. And these were available through the Kids First DRC Center, and we accessed that, and they are now accessible to us through Cavarica. And I'm just going to show you more or less how we found this data and how we got access to it through Cavarica. So on the Kids First DRC webpage, it's where I was looking what the data sets were available in there, and I found these 2 data sets, and we're trying to plan the experiment. And then if you press in each one of them, you can find access form, and you can fill the form. And then once that's done, the data was here accessed in our Kids First page for us to do the analysis, to look at the data. And this is our own data here also available to us. And if you go here to file repository, you can then filter the files, because not every file is related to a patient with glioma. So we actually, and not every file, not every patient has sequencing available. So in here in Kids First, we actually did all the filtering of what individuals, what files we would like to work with. And after that is done, we actually had to do a little bit more filtering, and that was not possible to do here as easily. So we exported the data to this TSV to continue filtering the files that we wanted to analyze. And then this is going to be analyzed actually in Cavarica. So there was this step that needed to be done in Kids First to select the files that we wanted to analyze in Cavarica. So that's where our data is now in Cavarica. The data is here, harmonized and ready in our own project called RO3 here to be analyzed in here. So that was that was not actually a very difficult process, with the help from the group that we are working with as part of this pilot. Azia, Jack, Renan was able to do that relatively easily. But then we started trying to get the few samples that we had in NView as part of sequence, as part of the Baylor Hopkins, also in Cavarica to be analyzed. And that's where we are still working on. This piece was not completed yet. I just wanted to show you what we found. It's easy in NView, and why we could not get our data in Cavarica, access through Cavarica yet. So if I go to NView, I can go to this exploration, and very easily here, there is a tab that says export to 7-bridge. And if I have access to the data set in here, I can just select this data set and export to 7-bridge, and I will have access through Cavarica. So here, export to 7-bridge, and you have here, export to Cavarica. So, but what we've found is that our data is not indexed to be accessible through this path yet. So we cannot actually send our data as we did that as a pilot here with the 1,000 genome, and we were able to have it here, the 1,000 genome test in Cavarica, to be accessed and analyzed in that together with the other samples. But we don't have it yet because our own samples that we sequence through the Baylor Hopkins are not indexed to be available to that export. So we have been talking to the group for NView and trying to figure out what's the best way to do it. Sounds like so far the best way for us is to wait for these data to be indexed so we can send it through that path that is just export to 7-bridge, export to Cavarica path. That's the easiest way to do it, but we don't have our data indexed there yet. So I think that's it's a quick overview of our project, where we are so far. We have now access to the glioma samples, and we can go on with our analysis and board analysis for the glioma samples, but we still want to access the chondrosarcoma samples, and we do want to bring together the samples that we sequence through Baylor Hopkins that are in NView to Cavarica also, to have access to everything through Cavarica and do the analysis through Cavarica. So I think this is what I wanted to show, and I would like to thank very much Azia, Jack, Jay, and Erica for helping us with this pilot project, bringing this access to all these samples in different places so we can do our analysis. And also Ruchi and Rachel from the Broad helping us with accessing things in NView. This is all I have to show today. Thank you.
Thank you, Nara, for this really nice presentation. I realize that we're falling a little bit behind schedule, so maybe we have time for one question.
This is Adam. Just a quick, maybe naive question, maybe on a Terra and Seven Bridges side. An alternative to the sort of export to Kavatica, I thought would be to just identify the DERS URIs for those files, and that might be another way to simply bring in those data into Kavatica. Am I wrong about that? Even independent of indexing, or?
This is Ian. Adam, I was just typing something about it in chat. Yeah, it's possible. There are a couple of different approaches though on how you use DERS as to whether you get the signed URL or whether Kavatica or the other Seven Bridges platforms use the DERS ID from within the platform. But if you get the signed URL and you pass it in via WES or by other means, then it can certainly access them.
Well, this is Michael Bauman. If I understand the issue that was presented here, the data it works with the open access data, but the data for the study, it sounds like, has not been indexed into Gen 3 yet. So it's not even available to explore and then hand off yet. So it's just a basic issue of data availability. Once it has been indexed as described, it can be handed off to Seven Bridges. That will import the DERS URIs. It could also be handed off to Terra, which would import both the DERS URIs and all the metadata, the tabular data that's associated with the study, and that would go into the Terra data tables all as part of the handoff.
So thanks.
Actually, the Ruchi and Rachel from the Terra is not here. They have a few more slides talking about the future plan to address this issue.
Yeah, there are some additional issues there too regarding the Anvil data currently being requester pays, which Terra supports, but Seven Bridges doesn't yet. So even when the data is available in Anvil, when it has been indexed and is available in the Explorer, and the DERS URIs are handed off, the fact that it is in request, the DERS URI is resolved to references to requester pays buckets, which Seven Bridges doesn't support. So that is another issue that I think was discussed with Rachel and Ruchi as well.
Yeah, I think, it might be good for one of the breakouts. Seven Bridges doesn't support requester pays because DERS doesn't support requester pays, and that's where we're getting blocked. But I think Ruchi put together some great slides. I don't know if we could move that to a later time. I know we're behind schedule.
Yeah, well, we use DERS to access data by requester pays, both natively and also now by signed URLs. It does require some additional setup, but it does work.
Valentina has a question. She's asking if only the Gen 3 index data in NView is available for export to Kavatica. And as far as I understand, yes, right?
Yeah, the GTEX data is made available in a special way by Gen 3. It's actually hosted on-prem and made available by CleverSafe, and can be accessed that way in an egress-free manner. But that's the only Anvil data at this point that's available without requester pays.
Okay, I appreciate this very nice, lively discussion. There's a lot to cover here and a lot to unpack. I appreciate your presentation, Nara, and maybe we can dive into some of these things a little bit deeper during the breakout discussions today. At this point, I think it's important to move on to the NCPI working group updates. We will start out with a the first update from the community governance working group and we'll hear from Bob and Stan. So Bob, you want to take it away?
I do, and thanks for advancing the slides. I have a poor internet connection. We're going to break this into two parts. We'll give a very brief update, Stan and I, on what's been done to date, and we reported it in the last quarterly, so it's just a few minutes. And then we'll talk about current activities over the last few months, which is on a new topic. Before we do that, I want to just remind everyone of the framework for this group. It's important that this group is not charged with coming up with specific policies or recommendations. That's just not what we do. That's what the institutes and centers do. Instead, we're identifying use cases and questions that can help precisely frame certain fundamental governance questions that either limit interoperability today or can encourage interoperability tomorrow. So our focus is on concepts and frameworks, and then through the calls and meetings like this, bringing the community to consensus, so at least it's clear what the research community, at least the research community that's part of the NCPI process specifically, feels are consensus items. And then we summarize the key questions, associated frameworks, and community consensus in technical papers. So, this is very important. We don't do policies recommendations. We identify use cases. We introduce frameworks, and we drive community consensus. And the outputs are white technical papers. Next slide. So we're going to talk about the first phase, in which we really focused on each of the, at least today, all the core NCPI platforms follow NIST 853 security and compliance. And we asked, when platforms follow NIST 853 security compliance, is there a simple process, simple consensus for interoperability? Next slide. So we simplified after quite a bit of discussion. There's the key concepts is there's a project sponsor that is typically an NIH institute or center, but it could be in certain cases, another organization. They are responsible for data governance, platform governance, and consortium governance. We call that the project sponsor. There's a cloud platform. The one cloud platform has the right to distribute data. This is determined by the project sponsor. And then another cloud platform has the right to receive data from a platform that can distribute it. In that case, if a particular data set can be transferred from a platform that can distribute it to a platform that can receive it in a particular environment, we call that an authorized environment. So the project sponsor for each data set determines what platforms have the right to distribute and what platforms are authorized, can provide authorized environment, it can receive the data. Next slide. So this is the picture. That's a happy project sponsor on the left. They determine the one platform on the bottom with the right to distribute, another platform at the top with the right to receive it. And we have interoperability in this context, and data could be in one platform and move to another for a particular authorized user in a particular authorized workspace. This is our review. Next slide. This is summarized in a short paper that you see there, the consensus. This is the technical paper. We introduced the term SAFE for a secure, authorized, fair environment, but just so that we could have a slogan towards fair data in safe environments. And we're in the process of producing a slightly longer white paper that gives a little more background, makes this a little easier. But this is work that's been done to date and is review. Next slide. Next steps are where we are today is, as we've heard, for certain data sets, they can be moved between selected platforms, but I don't think at this point, at least as far as has been discussed on our calls, whether we have blanket interoperability between two NCPI platforms. Next slide. So next steps would be to try to move towards some more robust interoperability in the context of NIST 853 for the NCPI platforms. Next slide. So the last few months have been focused on another topic, which is, if it's a challenge for interoperability for two platforms with NIST 853 for standard controlled access genomic data, perhaps there are cases that we can move forward on in which, for various reasons, the data may be slightly less sensitive. And so we looked at interoperability when the data might be less sensitive. Next slide. And this is the observation that a number of people have made that, for example, certain aggregate data or certain summary data or certain data after certain authorized workflows may be less sensitive, and it may be easier to support interoperability for this data. Again, where we would still have the core framework of a platform has the right to distribute data, and the authorized environment would still have a formal framework for authorization, such as FSMA low or CUI, and we'll talk about that in a minute, but maybe it might be easier to distribute less sensitive data for interoperability. Next slide. So this is the same picture, but now the data is low sensitivity. We're just going to have standard sensitivity, which we call moderate. This would be the kind of controlled access data that each of the NCPI platforms has today. But there may be certain data, for example, derived data from a workflow or aggregate or summary data from a workflow may be declared less sensitive by the project sponsor. And we ask the question, could it be moved to a cloud platform B that may be authorized for CUI or FSMA low to other compliance regimes that are a little less burdensome than FSMA moderate? Next slide. I'm going to say a little bit about CUI. It's a formal platform. It follows NIST 800-171. It's not designed for sensitive genomic data, at least it's designed for less sensitive data, and it has policies, procedures, and controls that follow FSMA, but follow policies, procedure, and controls that follow this special publication 800-171. I misspoke. It follows 800-171. Next slide. So to test whether we understand this, we're trying to take the world's simplest use case, and if we could build consensus around the world's simplest use case, then maybe we can move forward. So the next slide, please move forward, has the world's simplest use case. This is a slide. This is a use case in which we're in one FSMA, we're in one platform that has it follows 853 like the current NCPI platforms. There's data, say genomic data that is in this that has let's call it moderate sensitivity. There's a workflow. The workflow might be some summarization, might be a GWAS, might be any of the standard workflows. That workflow is vetted and approved through some process approved by the project sponsor. That workflow is locked down, so it can't be changed. And then that workflow is retrieved. And again, it's checked that it has not changed since it's been approved. It's applied to the moderate sensitivity data, and by consensus, when applied to data of moderate sensitivity, genomic data, through some process, the output data is lower sensitivity and maybe, for example, put into a CUI platform following NIST 800-171. And then if there is such a platform, let's call it platform B, which is authorized for CUI, then presumably there could be a transfer since we have the right to distribute, in this case, low sensitivity data, an authorized environment to receive it. And we have the broad characteristics that we've had before and described in that white paper, but it is now for low sensitivity data. Next slide. I want to pause for a question. I'm going to go through the rest of the slides a little more quickly so that after this one, so we have time for questions. But you know, the questions we have are the same questions we have whenever we move data from one platform to another, from one cloud platform to another. These are not easy questions, but the reason we think through these is we know that when data is shared through authorized cloud platforms, there's acceleration of research. There's the ability to do research that might be quite hard if you can't combine data from two different platforms. And there's a benefit to, you know, to the human subject. So for all those reasons, we have to look at certain questions. They don't have easy answers. For example, if there's a data event or a security event or a data incident or a data management incident or event, who's responsible? Is it the platform that receives the data? Is it determined by the platform sponsor? Is it determined by an agreement? Is it determined by a memo? Is it the platform that sends the data? Is it depend on the specifics of the event or incident? In practice, are the answers different if the authorized environment and the platform that has the right to distribute are from the same institute or center, or when they're in different institutes or center? You know, in practice, it's some complex combination of all those, and that presumably is what's made the progress a little harder, because there's no simple right or wrong answer. It's a complex series of trade-offs. And you know, you know, even though we have certain data sets from two platforms that have you know, one-off approval to interoperate, you know, people tend to answer these questions conservatively, which is why we're trying to ask, would it be a little easier if the data is less sensitive to be a little more liberal in how the questions are interpreted? Next slide. So there's a, you know, there's no right or wrong answer. It's a trade-off between benefits and risks. Benefits are acceleration of research, research that couldn't be done otherwise because the data is in two platforms, and you know, and related, and you know, the associated human impact. Risks are the you know, incidents related to security or data management. There's a standard framework I'm not going to discuss, but we are looking, you know, we are proposing to use this, and it's referenced here. Next slide. So we're not trying to introduce anything more. It's, you know, the risk is the impact versus the likelihood. The benefit is the benefit as measured, and it's not easy to measure benefit of research. It's not easy to measure acceleration. You know, we have frameworks such as DALI and others that can sort of do this, but it's a lot of work to do that, and perhaps we need more work on that. Next slide. You know, you can enumerate the risks in this lockdown example. You know, you know, someone may have spoofed the Docker, or the person who gets the data may inadvertently expose it. On the other hand, you know, it is lower sensitivity data. So next slide. So, you know, we would formally look at each risk, look at its use case, talk about mitigation. You know, these are tough things to do. I'm going to pass it to Stan now for some summary comments and discussion of next steps. So next slide.
So Bob's covered the ground thoroughly. So I guess the right thing to do is for us to take questions and answers or comments. Bob is always masterful.
Bob, this is Ian. I had one question. And I'm not sure I can follow this through everything you've said, but maybe you can help do that. So a lot of what you've been talking about, and I think we still use this language a lot. We talk about transfer and download of data. But in many cases now, that's not what we're doing. We're leaving the data where it is. And what we're doing is, one might say, transferring or uploading the compute to the data. And that's kind of the model for many use cases that's preferred. I suspect, though, that a lot of the issues that you're raising still apply in that. But I feel like we should be thinking that use case through as well. I don't know if you have any comments on that.
Yeah, I think that's important. The way the current document is written, I think formally covers it because there's simply two cloud platforms, one where analysis is permitted and the other where the data is. So for example, if the authorized environment where analysis is permitted wants to ship a container, that's perfectly permissible and covers, what you might be talking about. On the other hand, if the data, the source, the first platform has the data and the analysis platform wants to access that data, like we've heard through DRS, that's permissible if it's an authorized environment. We thought we had a number of discussions about what the next use case was. We chose, working with less sensitive data. But Ian, you obviously raised a very important issue. The other thing we thought about is this idea of basically shipping the analysis to the site. And if you look at that use case carefully, it was motivated by precisely what you just said, Ian, in that the primary motivation was the other platform has an authorized workflow. It's put in a repository and it's executed in the next to the data in the platform that has the right to distribute the data. The output of that analysis is then has to be returned for some kind of aggregate analysis to the platform, to the authorized environment. So I should have emphasized this, but when you are shipping containers to be executed close to the data, you still want the aggregated results returned, the summary results returned. And so the use case we did, even though we didn't explain it explicitly in this context, is designed to support remote execution of containers next to data and return results, as well as federated execution and return results for central analysis. We're assuming the results are less sensitive for the reasons I described. But what you just described was one of the primary motivations, but we didn't explain it very well.
Thank you for this nice conversation, Robert, Stanley, and I apologize that we have to move on, but the time is tight. So Jack, do you want to take it away?
Yeah, thanks so much, Stephen. Can you hear me? Awesome. So Jack DiGiovanna, Brian O'Connor and I co-chair the System Interoperation Working Group. I'm delighted to present some of the progress that we've made and things that we focused on as a group over the last 6 months. One question I got, though, despite we here in this audience, I think, see the promise of interop quite clearly. I think the excellent talks from Melissa and Nara earlier show that. But folks came to me and said, well, why do we care so much about interop? What's the point? Why focus on this and not just build a single solution? And I wanted to frame this here was like looking at the budget requests from the National Cancer Institute. They each have highlighted scientific opportunities. 2022 is on the left here, 2021 is on the right. And to me, from a mission perspective, and I suspect for a lot of us in this room, this is what's exciting about this field. This is what brought us in. This is a chance to make an impact in precision medicine, to change the lives of patients and their families. But in my prior job, I was also doing machine learning. In my machine learning hat, looking at this, I say, well, this is really requiring many things, but one of the things it's going to require is bringing a lot of data together. These data sets aren't sitting inside of a node that we just need to unleash a master student on and have them be able to drive these things, but actually to be able to interoperate between data where it lives. And that is the mission of the System Interoperability Working Group. So empowering more researchers to be able to attack these kind of scientific questions. And along the way, spearheading technical improvements so that when we solve this for the first researcher, it's easier for the next 5 researchers who come through so they can be faster speed to results. So System Interop is part of many different working groups within NCPI, and Bob and Stan just gave a great talk about governance. We overlap a lot with them. We overlap with FHIR, search, outreach, and training, but we're only covering part of the researcher journey. So at the start of the researcher journey, probably through FHIR or search or one of the great portals that have been shown or divine intervention, the researcher understands this is the data that I want to analyze and explore. System Interop is here in the middle, kind of the plumbing, the boring part, but making sure that that information can be formatted in a way such that when it gets passed over to a workspace, the workspace understands what the information is, and the workspace can put that information in a format such that the researcher can continue along her analysis journey and get to the analytics and get to the results. And abstracting that out here to a diagram, so going to explore and discover which data is relevant, then handing off that result to a workspace, of course, checking off Z at the time of analysis to make sure that the user is authorized to it, and putting the results together so that the researcher can analyze it. And one of the earlier use cases recently published, which is really exciting to see, in this case, they were working with 2 data sets, a data set within the GDC and a data set within the PDC, bringing those 2 together on the Cancer Genomics Cloud within CRDC. And then looking at line 1 expression, Wilson wanted to look into healthy controls. So they brought in GTEx data from Gen 3 and Terra and Anvil and brought that onto the CGC. And we're able to bring that together, and that's really exciting. And the other thing you may notice on this slide, though, is that these ecosystems and these 2 ecosystems were created before NCPI. So one of the things we ran into in System Interop is some butterfly effects here. We're bringing that data together, as Bob just nicely introduced. There are differences in the improved environments between CRDC and Anvil. And not to say one is better or worse, but to say that this was a policy issue that we ran into as we built the technical interop. We then ran into, well, policy-wise, can we do this? But we worked together between the Seven Bridges team, the Broad team, the UChicago team to create interconnect security agreements so that now future researchers, unlike Wilson, could benefit from that and be able to bring data from Anvil over to a Seven Bridges platform, leveraging one of these ISAs, and made it easier for the next researcher to get things done. Brian and I have done this, and the working group has done this by agreeing on a finite set of technical methods. I think Titus gave an excellent talk yesterday on DERS. And we've not just adopted DERS within System Interop, but we've also worked to push it through issues or pull requests for aspects of the spec that were not quite meeting what we needed from real-world integrations. A number of them are listed here. One we're currently working on is the name as a required field. We've also partnered with NIH RAS. So a lot of the different groups that were involved in this were also in kind of milestone 1 for RAS and working together with them, working through the processes, working to get all the forms and things right, where a lot of us are establishing now the TLS certificates that are required for the different servers. And we're working together with RAS and with the NIH to look at how many passports do we need for the different servers and how can we resolve that challenge. And then finally, how is this information transmitted? It's through a manifest. And initially we pushed very hard on PFB, an Avro-based format, and that worked really well for a number of workspaces and portals. There were some sticking points where other portals had CSV exports, and that was the only solution. So Brian and I earlier in this year with the working group decided we'll allow for either PFB or CSV, but the important thing is what's in them. So what are the minimal metadata attributes here? And importantly, this is not a data model. I don't care which data model you put this in. The important thing is that somewhere within that model, the workspace can find DIRS_URI and not URI or DIRS_ID or concatenated, right? Because importantly, we need the workspaces to understand what information is coming over it. We're currently working with the group to review these attributes and whether they should be required or suggested. And currently the only required one is this one here in orange. Credit to Asia Lin, who put together a minimal criteria, which I feel has dramatically improved the use cases. They created a one-pager that ensures that a certain set of info is agreed upon. This is based on experience we had in the first years of the project. And then Dave Rogers has put together a really nice use case tracker here with the link down here on the slides, which is showing, oh, sorry, I have the camera over, but I trust that it's still showing system interop, search, FHIR, the status of things, and you can link into this, get into any of the one-pagers, and additionally, what we've seen, I mean, looking at the different use cases, Nara's, for example, I think only came into this in late December of last year, but by having all this information put together, because Asia designed this really well, she was able to move forward very quickly because we didn't run into the stumbling blocks. There were still scientific stumbling blocks and access issues, but all the things that we've learned in the past, we were able to leverage to be able to move forward more quickly. So overall, I'm pretty happy to see how things have progressed. I'll say personally, as a co-chair, and I think I probably speak for a number of other co-chairs in NCPI, early in this effort, I really felt like a traveling salesman, going to researchers and saying, gee whiz, I know you already finished your analysis and you're trying to write your paper, but could you please rerun it? Because I want to benchmark this for system interop. Or I know you have this manifest, but could you try that manifest? Or I'll write the script. If you just run the script, I'll write it. And that was, I think, necessary to get us started. And a lot of people helped to do that, but it really not sustainable. And the funding and roadmap management was also challenging, despite really great support from ODSS. But today what we're seeing is use cases publishing manuscripts and more interop is happening. This is an aside, but the CFDE, the RADX, the Include project, none of these are formally within system interop yet, but there is a nice cross-pollination of people who are in this working group and who are in those projects. And the way that those interop is getting designed in those projects is leveraging a lot of the principles and lessons learned. So it's happening faster. And more than that, it allows for a future when you want to, you're integrating currently with Workspace A, you'll be easier able to integrate with Workspace B because Workspace B already knows how to talk to NCPI things. The other thing that made me quite happy was on the tools and data set side. So we meet every 2 weeks, and one meeting a few months ago, we were talking through use cases, just where we were, and everybody talking about their things. And it was, Melissa was talking about her use case and different things that they're trying to do. And the room was just getting excited. And out of nowhere, Mike Schatz unprompted said, you know what? We've just made this tool in the T2T consortium and it's optimized, it's available on WDL. Do you want to use it? And similarly, Jay Ronquillo, an NCI data scholar, was talking through his use case and the different data sets that he wanted to use. And Adam Resnick jumped in and said, oh, that's great. You know what other data set would be great for this? This Kids First data set, which would complement that. And seeing the community help each other and be able to do things faster is really exciting. The other aspect in the beginning, I said, in the mission statement, we want to make this accessible for all users. Now, unlike yesterday, users farther away from the engineers also have to be able to do this. So if you put scientific expertise on the Y and tech expertise on the X, initially some of the early results were very much unicorns over here. And, I got called out as only very technical people who can write JSONs are doing that. And that hurt my feelings a little bit, but I'm excited to see that we've actually moved to the left. We still have room to improve. We still have more users to enable, but the types of integrations that I think you saw in the prior 2 talks are very much, if you can go on, find a data set and push a button, it will appear in the workspace of your choice. And that's really solving for more people with less technical expertise to be able to do it. So summarizing, really appreciate all of ODSS's support and partnership. It's exciting to see how we're increasing speed to results through the things that are happening in NCPI system interop. We really want to build awareness and adoption because, for about a year now, researchers have been able to analyze select CRDC, TopMed, Kids First, and AMBO data within either a Terra platform or a Seven Bridges platform. But the real call to action here from Brian and I, please connect with us with the latest research or challenges. We want to know what to do next. We want to know how to adapt our strategy and what would make the most impact for researchers and for the field. Thank you very much.
Thank you, Jack, for that nice talk. Okay, if there are no questions now, we can approach Jack in the breakout sessions or elsewhere. I appreciate the talk again, Jack. Let's, at this time, let's move forward with our next talk by the FHIR working group. This will be, this is chaired by both Robert Carroll and Allison Heath. Take it away, folks.
Hi, Stephen. Thank you. I'm really excited. It's always fun to be in the middle here because I think you'll hear very similar themes from the work we've been doing with the FHIR working group. Next slide, please. So I've got 5 overview items I want to talk about. We'll talk about our objectives, some of the services we've deployed right now, the work we've done in the implementation guide, what we're doing next, and then an exciting event we're hosting next week. So next slide, please. So the objectives of the FHIR working group and what we're looking to do with these services. This is a slide I've used before, some of you may have seen. Right, is this idea to provide an API to allow access to study and participant level data, to provide those standardized structures for study and participant level data, and to enable structured semantics for the data where available. And I think this is trying to really build in collaboration with these other services we're talking about, where things like DIRS as a service can go hand in hand with these with the ideas of FHIR, for example, where we can really provide those structured semantics. And so that's the goal, that's the objective that we're really looking for. And we're benefiting from the sense that a lot of cloud vendors, for example, are providing tools, managed cloud services to actually support this. So we're really hoping to leverage some of the energy that exists, particularly because of the healthcare community. Next slide, please. So at a high level, and I put other working groups in here, and so I'm not speaking for them canonically, but I think it's important to consider how the work might play in together. And so as the FHIR working group there, that big box in the center, we're building an implementation guide with a set of tools talking about data on a participant level, at a study level, and how that might interface with some of the other working groups, particularly some of the solid arrows there. Right, we have file metadata and we're using DIRS URIs. So we're really crossing the boundaries there among the different services and the different standards that we're working through in NCPI. And our goal then too, right, is those ideas that workflows then could actually have a structured semantic way to get into some of those really the deeper level of participant level data, right, that I think a lot of times is kind of trapped in these tables without clear semantics. Additionally, groups like the search working group, right? And to be clear, these aren't necessarily direct API calls like that they need to rely on this like on a query basis, but to give something like a search working group a standardized interface so they can see what data is represented across these platforms. In that sense of, hey, what vocabularies, the data dictionary type of information, right? It's scattered and it's not necessarily consistently represented. So if we can help provide an interface to make that information available, right, where we're reaching out and we're working with those same vocabularies and ontologies that the search working group might be interfacing with. And so we're really trying to leverage, again, those external standards to really do our work. Next slide, please. So I wanted to cover the FHIR services that are available right now. These are ones that I'm aware of, so there are others out there. So please, I hope you don't feel excluded if you're not on here. The formal NCPI teams that have deployed some services that we'll be using very soon and are out there right now. Kids First team has production FHIR services deployed. You can go access them right now. A lot of the clinical data is open access, so you just go register and then you can actually work with a lot of this really neat clinical data that's available. And again, that does also reference you right to those DIRS URIs. And if you have access, you can get the files. If you don't, the DIRS is going to shut you out at the door. So that's really exciting. I encourage you to go check that out. dbGaP has some public services deployed. They've got a first pass implementation on the study level data that's publicly available. Anyone can go hit this URL and go visit. Additionally, they are doing work on the controlled access data. It sounds like they've got some pilot implementations available to internal parties only. I think it's within the NCPI firewalls and that kind of thing, but they're doing a lot of good work over there. The Anvil team, we've done a number of non-production service pilots. We've got Brian Walsh that's really neat work deploying some services, the index data that's available across Terra. So leveraging those Terra data tables as well as those DRS references. And my team's done some work, Eric Torstensen, on piloting study-specific ETL, actually doing the transformations really with those detailed clinical and phenotypic semantics and connected with the data. So we've got some really cool work, and we're hoping to continue that into the future. There's a couple of community groups I wanted to highlight in here. And so they Import, for one, is Ben joining us in the FHIR working group. The Import is not an official NCPI member, but we've been really excited to see the cool work that they're doing. They have a whole IG. They've deployed services. This dev service that I've linked here is open access and available, but I think they do have some controlled access services as well. Additionally, the include DCC, again, they're involved with NCPI. They've got some use cases spun up. They also have a production FHIR service that has registered user data access. So again, once you sign up and agree to those the data use agreement, you can actually go in and access a lot of the include specific data as well. And so that's really exciting. It's quite easy to get into, which I think is a big change from some of our prior presentations where we didn't actually have that data, the real world data that everyone get access to. So like I said, there are a few data sets here. Please feel free to dive in. Next slide, please. So in the past, we developed this original implementation guide published last year, and we've been continuing to do some iterations. We had a really strong focus on some of the rare disease modeling because the semantics are pretty strong and clear. But we've also done a couple of these live deployments now, and we've got some really valuable feedback that we're looking to kind of really iterate on and clarify the spec. And that's making some broader use cases and refining some of those approaches to asserting semantics. There's a lot of tension. Again, Jack was like, we're not defining a data model here, right? And I think that's always one of those big challenges from, keeping with the original study data versus these harmonized, standardized terms. And so we're really looking to make sure how can we, not necessarily say this is the only expression of this data, but to say we're going to link to these semantics that everybody knows are important to these core entities. And this is how you would do it in FHIR so that you can get these benefits of these services. And so next slide, please. That's something that we're working on both in communication and the technical specifications. So we've got right now, we in the past had this cloud of things that you might want to put in there. And again, this is about that set of services and metadata to really for the data and metadata to apply to the researchers. And not everybody has those. And that's one thing we ran into with, again, the rare disease. A rare disease and a rare disease phenotype have really specific meanings, but they don't necessarily work for everybody else. So next slide, please. So the goal then is that we're working on is to reorganize this in a way that you can talk about clear modules, and we can say, hey, this study supports rare disease. Maybe this other one doesn't, and that's okay because it still has the genomics files. And so really trying to clarify and anchor on those use cases. And this is rough, this is early, this is for demonstration purposes here, but to really kind of show what might a service want to provide. If somebody wanted to interact with NCPI, it's not here's a sea of things you could do. Here's a few set of core use cases that drive what you might want to adopt. So that's what we're working on now. Next slide, please. And so that's this next version of the IG. We've done some early reorganization, both on the documentation side, and we're working on continuing to improve the technical implementations of those definitions. And yeah, I think the big thing, want to make this documentation more clear, more accessible to users. So I'm happy to get feedback from anyone on that. Like I said, it's a work in progress. It's exciting. Next slide, please. I think that brings us to the last slide here. I think next to this event, we have next week, Monday and Tuesday, we're going to pull a let's say lightweight, but a real world use case together for a codathon. We've got some good engagement and buy-in. I've been working, hard over the last couple of weeks on documentation for this. And so to help people get connected. And so we'll be really doing an end-to-end real-world analysis, taking RNA-seq gene expression data. Fortunately, a lot of this data is open access. And so we'll actually be using open access GTEx data from Kids First, data from Include that are registered access that are really easy to get your hands on. And we're actually going to do using DERS, using FHIR, hopefully using workflows, if we get the right expertise to join us, so we can actually do some of this work, which I think really aligns with what Titus was talking about yesterday. What Jack was just talking about is that we can use these actual services to do this in a really consistent manner, hopefully across these different representation or across these different services provided by platforms. So I think it's super exciting. I think we're actually there. This stuff is live in some cases. Again, we've got some on the ABLE side. We have some, development sort of pilot just for this project that we can share with people that uses open access data. But I think it'll give a really good picture if you're interested. We've got a set of documentation. And actually, there's one more slide. Get the next slide, please, Stephen. So here are all the links. They're in the notes and the slides. Registration link if you're interested. I've got the GitHub repository there has some introductions, like how do I get access to Kids First data? How do I actually query these FHIR services? Helping with the authentication pieces a little bit. So we'll continue to develop that. And that's, again, we really hope to get some technical support, scientific support, there's documentation support. There's a lot of to really help people get up to speed. I think technically we're right there. It's really close. So if we can bring all these three aspects together, I think we're really, next time we meet, we'll be able to show off a lot of the cool work. So, and again, if you can't make it, that's okay. Keep an eye on these repos, and we'll be sharing more in the future. So I think that covers it. Allison, did you have anything you wanted to add?
No, I think that covered a lot of it, and I think really excited about doing more of the implementations and really running different data sets through. And I think as you mentioned, the codathon being this kind of layer on this overarching use case that we've been hearing about across GTEx and Kids First. So I think that's all I wanted to say. I think you covered it nicely. And it's been really nice to see the working group kind of move back into an implementation phase again.
Thank you both for that nice presentation. We might have time for a question if anyone has one.
I've got one question, Robert, and you sort of said it in what you were saying. You almost have to do this. Oh, no, I'm not creating a data model here because there's some pressure not to create a data model because there's a standard data model that we should be using, the FHIR data model. But I think, and I think this came up in the search group last week, and I think maybe maybe we're going to hear from that shortly, and maybe this will come up. But we got the use case in research that we need to create data models because the structure of a scientific study is a unique data model, and one of the questions is how we fit that into FHIR. And I'll keep it short too. We had some, this is from a CRDC point of view, had some discussions last year with the BR&R group within HL7 and recognizing that use case. And it became a question of, is one allowed to do it within FHIR? And I think that's, how one would do it within FHIR is through what's called I believe it's through the FHIR Foundation model on which everything else is based. But the useful term that came up in search last week, or certainly in the Slack afterwards, was meta model. And standardizing on the meta model, and there's a whole can of worms there, which isn't time to go into now, but standardizing on the meta model and allowing different data models is maybe a way where we can get an enormous amount of leverage.
Yeah, Ian. So I think that is one of the reframing, thanks for bringing this up, that we're trying to do a little bit, is again, trying to get away from the sense of, this is not a general case prescriptive, like you must, or prescriptive data model, like you must do everything this way. It's to accomplish a specific set of use cases, these are the attributes you must define. These are the semantics that have to be there. Here's how we're defining in the FHIR context. So if I want to provide a service that allows me to look at rare disease phenotypes, these are the attributes. These are how we're representing them in FHIR. And then we can use, FHIR includes this, LinkML includes this, there's ability that sort of linked data approaches where you can assign those attributes, like in the context of a specific implementation guide and a specific profile, you can tie those to that meta model you're describing and say, this field is a participant identifier as defined by an NCPI or an NIH or a worldwide research meta model. And so then you can have that linked data attributes. And so we can say, for the services we're going to provide, and maybe it's, again, a platform may not want to provide that set of services, may not apply to that set of data. Totally okay. The source data model may look totally different. That's great. But this is what you have to do to meet this specific use case. So that I think we're headed in that direction. So we can support that in a powerful way. But we're not going to block on everyone in the world has to agree on the right data model first.
But I think to the point, you also want to delegate and distribute that question of, here are the attributes you must provide. That can be federated out to different appropriate groups for different projects. Finished.
Okay, I hate to have to cut off this conversation, but we've got 15 minutes to get through 2 talks before we start cutting into our lunch. So I'm going to move us along. Thank you very much, Robert. Thank you, Allison and Ian, for your question. And at this point, we'll jump into the slides for the NCPI Outreach Working Group, chaired by myself, Stephen Mosher. So I'm going to do my best to rush through this and see if we can't minimize the loss of time through lunch. So the NCPI Working Group mission, as stated when I started working with this team, is to prevent the development of siloed platforms by providing unified access to key information and training resources associated with each NCPI platform. And for sure, all of the platforms are not islands. We need to work together. And so we have built up some goals to help try to achieve this vision. And they include developing and maintaining an NCPI portal, aggregating platform-related related outreach and training materials, documenting some commonly used resources, developing and maintaining a catalog of NCPI data sets, and support of NCPI workshops, which this is one up. So we have a portal for the NCPI organization, for the NCPI effort, and it can be found at anvilproject.org/NCPI. And when you visit this web page, you'll see some high-level ideas about what it is that the NCPI group is trying to achieve. I think that we've heard a lot about those today and yesterday, but there's quite a bit more information available on the portal as well, including the description of each of the participating platforms. And we've put together some high-level ideas or blurbs around some of the technologies enabling the science that is important to the NCPI effort. So this includes quite a few of the things that we've been hearing about over the last couple days, including RAS, DERS, FHIR, and PFB, the portable format for bioinformatics. And of course, these technologies are enabling science, and the science that's enabling the tech or driving the technology, it is listed out also on our demonstration projects. If you come to the demonstration projects tab on the portal, what you will be able to do is take a deeper look into some of the talks that we heard today, as well as some other ones that we did not have time to hear about. So please do visit these demonstration projects if you would like to have a deeper look into some of the work going on within NCPI. Another effort that we have going forward is aggregating and maintaining some of the platform-based outreach resources. So here you can find, if you have any particular questions about how things are done on one particular platform compared to another, this is a great place to dig in and start diving a little bit deeper into some of the how-tos, some of the tutorials, and some of the training materials available. You can also find some of the past, some resources from the past workshops. This is our sixth workshop, and you can identify some additional information, past agendas, past slides, past notes. Go ahead and take a look at those if you're interested in what's been going on. And of course, as I said before, one of the key things that is really, really important to what we're doing is knowing what is the data that's out there. What is the data sets that are currently available within NCPI? And currently we have 12 petabytes of data across 830,000 participants, and it's growing really, really fast. So to support questions of what's out there, what's available, together with the portal team and Clever Canary with Dave Rogers and the search working group, which we'll hear from next, there is now a dataset catalog, which lists out the different studies by platform. And this allows for some faceted searching. I'm not going to dive into this at this point to save time. I think that we might hear a little bit more of that from the search working group. I'd like to also point out that the DocStore team has put together an organization for NCPI, and this is a great way that one can promote FAIR practices and share tools and workflows. So I mentioned that this is the 6th workshop, and the NCPI group is, the NCPI Outreach Working Group is putting in a lot of effort to help support the various workshops as they come. And again, we're in the 6th one, currently being hosted by the Anvil team. And today's virtual workshop has received a lot of dedication from the Outreach Working Group, but also from the wider NCPI community and our partners to make today's event possible. It's been a busy schedule, a busy agenda. We've had 19 speakers with 15 breakout moderators, 8 note takers, 3 panelists, and 2 MCs. As of yesterday afternoon, we had 175 registered participants. I'm sure that by the time that I check later today, that number will have grown. So the last parting thought that I want to leave everyone with is the future of NCPI. The Outreach Working Group is really excited to see what's coming down the road. There has been a research opportunity announcement titled the NIH Cloud Platform Interoperability Administrative Coordinating Center. We look forward to working with the ACC in the future and driving forward some of the agenda of the NCPI effort. So with that, that is my last slide. I will refrain from accepting questions unless anyone's got something burning. Please put it into the chat. Happy to answer it there. And without any further ado, I'm going to pass things off to Dave Rogers.
Okay, thank you, Stephen. Thanks for the introduction. So yes, spoiler alert, we also will not be advocating the creation of a new data model. And we'll try to get through this quickly so that we can continue the discussion that Robert and Ian were going with about the meta model. So yes, I'm Dave Rogers. I'm a Clever Canary, and I am leading the implementation, the team that's implementing the Anvil dataset catalog, the NCPI dataset catalog, and a few other projects related to researchers accessing their data. And my co-lead, who will be or the search working group, is Kathy Reinold, and she leads the data modeling team at the Broad and is a champion of findability and data interoperability. And then I should mention that we have a close collaborator, Asia Lin, who was the NIH GRI data scholar for NCPI, and she's our close collaborator and advisor. Next slide. All right, so overview of what we'll talk about. We'll go over, this is a newly formed group. The search work group is the newest group that's formed. We formed just after the last meeting 6 months ago. And so we'll go over our mission, vision, our strategy for achieving our mission vision, the high level search use cases that we're trying to support, and then an overview of some of the work that the group has done. We responded to the ODSS search RFI. There's a RFI went out, and the group worked to come up with a team response. There were individual responses, but there's a team response as well. And then Kathy and I and the rest of the working group have been developing this search landscape survey. This is one of the first things that we decided should be done at our last meeting, and I was happy to see that Tanya mentioned a similar concept. Well, what's the current state? What's out there? What are the tools and platforms that we want to integrate, and what technologies and data models they use, and what challenges they're facing. So Kathy will go over that in a minute. And then we wanted to highlight our search demonstration project. We have a few search demonstration projects in the proposal phase, and then next steps, and then maybe some time for a discussion. So next slide. So I mentioned when we were founded. So we want to accelerate the improvement of search interoperability, across the participating platforms, and then have the shared vision of a trans-NIH federated data ecosystem. And then again, focus on data set discovery, cohort discovery, and knowledge discovery. And I was a little bit fuzzy on what is knowledge discovery. I think that there's different answers from different folks, but I think it's going over highly structured data sets with structured metadata and knowledge graphs, and using that to help find the data and or facts that you need. Next slide. And then more about the vision. There could be a lot of attributes on this vision list, but we want a data ecosystem that is, of course, fair. Federated comes up a lot, just like the internet, supporting data interoperability, and then transparency of provenance and data quality, and researcher and equity and participant equity. Researcher equity, I think there was a great slide on that in the last talk by Jackson showing, we want to be able to access the data. You don't want to have to be a platform programmer to access the data. And then participant equity, well, we want to make sure that we have, as NHGRI is, and many others are discussed, we want to make sure we analyze the genomes of individuals of all ancestries. So make sure that we include the metadata to know, well, what ancestries do we have? Is it easy to see? Is it collected? And can we tell what we have? And then how do we work? We advance our vision by identifying it, evaluating, promoting, and key to this is demonstrating the effective use of interoperability standards and guidelines. So we are primarily trying to find and promote standards and guidelines, and then also demonstrate them within our platforms. Next slides. Okay, and then just a little bit of clarity, the target use cases and search modalities, of course, by experimental metadata, what assay data type, study design. But the point of this slide is that there's a sea of, there's 4 big classes here, and a sea of different types of data or metadata to analyze. And it looks like there's standards sure obviously could help here, because if everybody represents all these different things differently, then searching over it coherently will be very difficult. Next slide. Okay, and then the team strategy is number 1, be driven by scientific use cases. So make sure everything we're trying to do is grounded in try to ground it in something that a researcher is actually trying to do. Common data standards and models. So we want to foster sharing and knowledge sharing across the community. So we're having our group meetings for that, and also the NCPI portal helps with that. And then another thing we do is solicit these and facilitate these search demonstration projects to provide, again, concrete examples of the standards and guidelines in action. And then another thing that's key, is this has come up before, is to help promote the best open access view of managed access data sets. I think Melissa Wilson mentioned this, like trying to know whether or not it's worthwhile requesting access for a data set, because in her experience, getting access was more complicated than it could be easier. Let's say that. Next slide. I'll also say that the mean time for getting access for dbGaP is pretty low. I think it's like 2 weeks or something like this, but there are many. It's a complicated. There's lots of complexity there. So Okay, so let's go into some of the work that we've done recently. And so I mentioned we did a RFI response, and then there's 2 slides on this. So we advocated for this, open and federated data ecosystem in the response standards adoption. Let me get to something that's more unique here. Yes, exploring FHIR as an API solution for representing the research data at the study and metadata individual level, not just the clinical metadata. So I think the group advocated for that as well. And then another one that I think is really important is investing in tools that enable, basically data to be sort of born annotated or born semantic. So all the way from, from the lab, the LMS, or all the way from the sequencer and the LMS, so that it just comes out already annotated with the correct metadata. So that this can and common data elements. So there's less work to be done to harmonize the data, in the future. So next. And then other recommendations was establishing this minimum study metadata. Again, this is, as was mentioned, not a data model, but a set of attributes and vocabulary for those attributes. And then we advocated that the data cataloging and data explorer code to emphasize reusability. Now, a lot of these projects, all these projects are open source, but it's very difficult to stand up, an instance of some other project in many cases, because there's lots of other information needed to do that. So to focus on how can we make it so that it's much easier to stand up individual instances of some of the software that's used for these explorers and cohort builders, and also to support, to emphasize that multi-tenancy is an appropriate technique here. Multi-tenancy being that many different consortia or data sets or data set generators can be held in the same instance of a platform. So and then let's see, aligning on a mechanism to support the pan NIH data set. So we can go to the next slide. Okay, which gets us to our landscape survey, and Kathy will take over.
So, hello, everyone. First, just a quick note that as a data modeler, I'm getting a little nervous about the fact that data models are getting a bad name here. I think that the meta model that Ian mentioned is the model that we need. And we in this industry sort of use the word loosely and call schemas and formats data models. So anyway, that's it. Okay, getting onto the landscape survey. I'm going to go quickly through this because I know I'm standing between you and lunch. So the purpose was to get a view of what the current search capabilities are across the NCPI platforms. So we got responses from Anvil Gen 3, Biodata Catalyst, Picture. I'm not naming everything in this list, but you can see that we tried to go across all of the different groups to understand what the search was about and how people were using it. Next slide, please. General theme was, what are people looking for? This surprised me a little bit. I hear a lot about cohort discovery, but we seem to be really focusing on data set discovery. Maybe that's just because it's the first step, but it's an interesting point nonetheless. Next. So, and data modalities, what kinds of data are people storing? So this is important because what we see is that most groups are storing lots of different types of data. So really important for us to keep in mind as we figure out how to search across all these data types. Next slide, please. A summary about the standards. Of course, we asked key questions about several of them. What kinds of data standards do you use to capture phenotype? You can see there's quite a list here. I think this indicates that we're not totally clear on what we're talking about when we're talking about phenotypes, and also that people are using ontologies as much as they can, which is actually a good point. Next slide, please. Genotype standards and other data standards, you see sort of the same tendency. There's really a wide variety across the number of respondents that we had. And there seems to be a little bit of a difference between how the definitions were used. Oops, we have a typo with the Miami being in genotype standards, I think. But moving on, next slide, please. So for non-phenotype data, really 3 responses said we're not even doing non-phenotype. Others, again, that wide range of things using a lot of different vocabularies. One observation that I would like to make personally, not on behalf of the whole team, but I think we should consider recommending specific ontologies for certain types of data to prevent this, total divergence. There are hundreds and hundreds of ontologies out there. Social determinants of health is something that people are really not capturing at the current time in their search. Next slide, please. So just some key points that weren't covered in the standards piece was key technology enablers of cross-platform and search and cohort building include the internet, of course, common terminology, open APIs, interoperable data models, FHIR API, and file and subject metadata. Key metadata for search, we really did identify a couple of core concepts or terms that we needed to agree some vocabulary on, and there were some common, search criteria across all of these different search approaches. For consent, most of the groups were looking mostly at open data, but beyond that, they're looking at dbGaP consent groups, duo consent codes, a little bit in the future, I think, RAS. And for security, there was 1 reference to RAS and 5 responses that are FISMA moderate and FedRAMP certifications, which tells me we got the memo. Let's move on. So what are the challenges? Lack of metadata standards, lack of minimal standard quality of metadata, APIs, standardized APIs, different groups bringing their own data dictionaries, and how do you deal with mitigating all of these different data dictionaries? Heterogeneity of data formats, lack of collaboration. We need better focus on the science. But again, changing the, how do we manage the fact that in this domain, the data and the type of data that we're collecting does vary over time. So an observation from me is that that didn't come up in any of our conversations. So next slide, please. A couple of next steps, and I'm going to turn it back over to Dave for, wrapping up and saying what we're doing next in the NCPI search working group. Thanks.
All right, so this for the landscape survey, the next steps are continue to refine their survey with respect to a little bit more detail on what data models used and the indexing methods that are used, and then publish the survey results on the NCPI portal. So doing that and trying to write it all out long form will help us clarify it. Next slide. And then, so we wanted to quickly go over some of our work, our demonstration projects. We have several projects that are in the proposal phase. You can see we've seen several times now a link to the use case tracker. So if you have a search related use case, we'd love to hear about it. If you have an NCPI related use case, that's also the place to go, any kind of integration use case. And then, we'll just go over these 3 quickly. Uniform search is one that has come up. If you go to one repository and you do a search with a certain criteria, and you go to another repository and you do a search with the same criteria, you would hope that you would get similar results when they're talking about the same data set, and if they claim to be indexing the same subsets of data that you would find the same things. So that's not always the case. There's lots of reasons for that. So there's a study proposed by Anne and the dbGaP team to try to take a look at this in a specific instance and see what improvements can be made. And then the FHIR is one of the search tools that is fairly advanced in this ecosystem. It covers variable search. It covers search over a lot of the modalities that we've talked about, from variable search to metadata search to genomic search. And so there's a use case being proposed to integrate that with, there are already many implementations of this in hospital systems and also in biodata catalysts. And so we'll be looking to add one of the one of the NCPI repositories or platforms to integrate with a picture. And then back to standards. Duo is an emerging standard, G4G8 standard for representing the structured data use. That's a refinement of the dbGaP consent codes. And so there's a effort underway to say, well, if we can map very closely the Duo codes to the existing dbGaP consent codes, we could allow search in the NCPI dataset catalog using the Duo codes, maybe in addition to or instead of the dbGaP codes. And so those are some that are proposed. Next. And then our next steps as a group is to try to recruit additional members, but then make sure that we have something. Initially, the initial members will be helping us figure out what we should do next, but also to recruit members from additional places, not just giant platforms like Biodata Catalyst, but what about? And we need to discuss this as a team. But in my view, is what about the data generating consortia? What about data generating labs? And how can we all the way down the data generation pipeline start to get members that we can get that help us define what it is that people in their type should be doing to increase their usability of the data that's generated. And then as I mentioned, solicit and recruit the demonstration projects. Let's see, and one thing I think that's come up is to drill down a little bit more on these data model descriptions. What are tools that can do data model descriptions? And then propose initial data model standards for discovery. So working closely with the FHIR and the interop working groups. So I think that's our next steps. And so the next slide is the question slide.
Thank you, Dave. Thank you, Kathy. Any quick questions?
Are there any long questions? Ian, should we discuss?
Well, what I put in chat, Dave, was what you just mentioned about dbGaP consent codes and Duo codes, because dbGaP consent codes we use to drive Duo codes when they set out, and it looks like they may be diverging, and so that would be a pity.
I believe they're very close, but I don't know to the extent that they're diverging. I know there's some difference, and I believe that it's small. I believe that about 90% of the dbGaP codes that for existing data sets have been mapped to Duo codes. But and also there's there's a little bit of I guess refinement that needs to be done as well on the dbGaP codes.
Okay, so then
go ahead.
Valentina posted that Jonathan Lawson at the Broad is doing a systematic mapping of the dbGaP and Duo consent codes. So we can follow up with him for the next step for search working group, I guess.
Yeah, that would be one of the outcomes of this demonstration project, is to show, look, they're very close, or whoops, no, they're divergent. I think they're very close.
I mean, that's reactive, though. I think there's a proactive approach we can take to this too.
Right, and I noticed some codes that are not in the duo, so I submitted to their GitHub repo, so I hope they can extend those codes. Yeah.
Okay, thank you very much for this conversations of the morning of day 2. We look forward to the sessions in the afternoon, and we'll take our break now. Please feel free to stay signed on. We'll start again very soon.
Thank you so much, folks.
Good afternoon, everyone. Welcome back from our short break. So we are looking forward to getting started on our next session, which is called the technical aspects of interoperability. We'll start off with our talk for our first talk of the session from Dan Stanzione, who has a talk titled "The Texas Advanced Computing Center as an Interoperable Cloud Resource for Biomedical Research." I'll stop sharing now, and Dan, you can take it away.
Thank you, Stephen. How's my audio?
It's coming through clear.
Okay, great. I'm in a hallway at the University of Michigan. Well, I'm outside another workshop today. Let's see. And slides are visible?
They are in the correct mode.
Okay, great. Thank you. So let me dive right in. And yes, I switched times with Nick Weber, messing up Stephen's order in his slides there because Nick had a conflict. So, but yes, so I'm at the University of Michigan today, where we can have workshops where it's only 95 degrees instead of the 103 or so it would be in Austin. So nice to see many of you who I know. There's many of you I don't know. So I want to just run through a quick introduction to TACC, and then run through sort of things we do to make stuff portable among us and other providers, and in particular the commercial cloud providers. TACC is, mostly known, we're the Texas Advanced Computing Center, and we're a research unit of UT Austin, and we're mostly known for the big machines, right? So we have Stampede 2 in my virtual background here, the Frontera. Both of these are large NSF supported systems that have tens of thousands of users. And we are currently in planning for our next phase of expansion through what NSF is calling the Leadership Class Computing Facility. And should Congress approve it, that would pretty much be our 2025 to 2036 round of machines coming up. So we expect to be here and have compute and data for a long time. So in talking with Michael and Stephen and the organizers, the charge for my next 9 minutes here is, they asked me 4 questions. Is how can you leverage TACC for biomedical sciences? What resources are available? What technologies are you using to ensure interoperability? And come up with some research examples. And I'm going to try and answer all 4 of those, but I'm not going to do it in that order. It's going to be completely random. So if you haven't worked with us in the past, and I know some of you have, we have about 185 staff. We have about 12 megawatts of data centers. We have across several buildings, multiple visualization facilities, a whole bunch of production platforms. We actually have the largest academic machine in the world. In the US, we have number 1 and number 3. We put out about 9 billion compute hours a year. We have more than 100 petabytes of data. We see sort of 15,000 direct users and 50,000 or so portal users. And one of those portals that I'm not going to talk too much about, but with Mike here, is we host some of the main Galaxy sites. So if you were a Galaxy user, you may be a TACC user and not actually know it. So what we fundamentally do beyond having big hardware, although that's Lone Star 6, our newest fully immersed system. So that thing runs completely immersed in a mineral oil derivative with the dual AMD processors at 850 watts a node. So there's actually 4 cabinets in the picture there, each of which runs at a little over 70,000 watts per rack. So at this point, but obviously we provide computing and data and AI and software capabilities to help people support their research. We like to think we provide the expert help to be able to use it, and that we provide it in the modes that people want to consume it, whether it's the traditional HPC batch or Jupyter notebooks or interactive or through the Galaxy portal or whatever it is. And at this point, I think it goes without saying that computation, AI, and data are almost ubiquitous across the sciences. But in terms of what we offer, just from a hardware perspective, again, we have a whole bunch of platforms. Depending on how you consume us, you may care or not care which platform you actually run on. But Frontera and Stampede, I mentioned, are sort of big NSF systems. Frontera through 25, when it'll be replaced by Horizon. Lone Star 6 is our newest one that just came online this year and will run through 27. We try and overlap these, so there's always systems there. We have more AI-focused systems, we have more cloud-focused systems, we have a Kubernetes resource, we have a computer science testbed. We have 3 classes of storage systems, whether you want performance, high-integrity archive, or replicated publication DOI style storage. Actually, 3 storage systems distinct from the ones inside the main systems. We have 5 tiers of storage, actually. In aggregate, we have about 16,000, 17,000 servers that give you about 75 petaflops of performance, and we have about 1/3 of an exabyte of data. And generally, if there's a big computation going on somewhere, we're at least some small part of it. Last year, we had a couple covers of science. We had a whole issue of computing and science and engineering. If you saw the Event Horizon Telescope announcement by NSF a couple weeks ago of the imaging of the black hole in the center of the Milky Way, about 120 million compute hours of that ran with us. We were one of a number of providers in that case, but lots of big computation, which in many fields has been ubiquitous. We have evolved a lot in the life sciences in particular. And since I'm actually at a data and cancer workshop today, I have my handy cancer slide in how we address cancer-related things through supercomputing, right? And there's obviously the modeling piece, and I try and have examples of all of these, the large-scale data analysis piece, the molecular dynamics simulations are what we've done for 30 years, really, in life sciences computing. But lately, there's quantum calculations, looking at proton X-ray interaction with DNA. There's clinical trial design and planning, and obviously a lot of AI lately. I'll give you a few specific examples, but that's sort of the range, at least in cancer, where we do work. I'm having trouble managing all the Zoom menus with the slide. Yeah, so here's one example with MD Anderson Cancer Center, looking at oropharyngeal cancers, so head and neck cancers in particular, where a deep learning approach was able to get pretty reasonable accuracy in doing the segmentation and identifying contours associating with the tumor. It was competitive with having radiation oncologists read the what was there, so that was an encouraging sign. Let's see if I can move forward here. This is a more traditional modeling problem, but looking to come up with, and this is with the Dell Medical School here in Austin, or where I usually am, or at least occasionally am. But to take not a data-driven approach like deep learning does to understand cancer, but to come up with mathematical models around the sort of fundamental behaviors. And in this case, they used a combination of MRIs, PET scans, X-ray tomography, biopsies, and built an equation-based model rather than a statistical model that had about 87% accuracy in predicting response to breast cancer in a single treatment. Again, there's always special cases where you don't necessarily respond in the first round. This is a very different use of TACC. Both of those were sort of traditional ways to use a high-performance computer, but this is one that we see a lot lately, and usually in conjunction with an instrument. So, and this is with UT Health, also down in Houston, but what used to be known as the UT Health Science Center at Houston, not to be confused with MD Anderson, which is 6 blocks away. So, but in this case, we're using the Jetstream cloud resource because we needed real-time results with an API that used to be called Agave, now it's called Tapas, but an open source API to do quantitative analysis of MRIs and do real-time analytics. And so we've used development kits that have been built over the years through Cyverse and other projects to do this, and we can turn around MRIs in minutes in this by keeping dedicated virtual machines to talk back on these things. Another way that we interact, this is one of the many portals, Galaxy is another, but this is one by Stan Wadowich, which is the medical branch in Galveston at UT. It's a drug discovery portal. This is sort of a docking and folding virtual screening proposal portal that we had up. We use these techniques pretty extensively in a lot of the COVID work over the last few years, but this portal goes back almost 15 years with Stan, but it's a web-based opportunity to do screening on a library of molecules. And in this case, we actually built the portal interface and dealt with the software side in addition to the hardware. So, and then we've recently, in the last 2 years, entered into a pilot program with MD Anderson in particular, looking at the mathematical foundations of cancer treatment and computational cancer in particular. And we've had a series of pilot projects. This is one between MD Anderson and TACC looking at personalized dosing of proton therapies. And in this case, we're doing mostly code optimization because we need, again, several minutes turnarounds. We're probably going to host this in a VM or something on site rather than run it on a big machine. But right now we need to accelerate the computation. So it's more of a software research type thing. So that, and then our biggest project within IH is the acute to chronic pain, where we're doing real-time MRI, protected health data storage, protected computing on personalized but not de-identified information across multiple centers. I think a lot of you may have pieces of this project or some of the related projects. And then I had to slip in one classical supercomputing PDE one. This is Ying Li up at UConn. Where he's working on these alternative structures for nanomedicine delivery instead of the usual spherical balls, these sort of squiggly looking things that they call nanoworms for obvious reasons that are a little better at wiggling between red blood cells and making sharp turns into capillaries. And they're also magnetic, so you can guide them to the tumor a little bit better. So there's really a whole range of things we do, and that's just a few of them. So so I wanted to take my last few minutes and just talk about things we do to help computations move around from site to site and to make things a little more portable, not only between clouds, but say from laptop to cloud. So containerization is a big part of this that we actively support now. We support Singularity, Charlie Cloud, Apptainer, a few other container technologies. The containerized workflows you would build elsewhere, you can run them at TACC. We don't actually support Docker because of some of the root issues around that, but if you push your Docker images into something like biocontainers or another repository, we can check out the recipe and run them as Singularity containers. At this point, that's just sort of good software engineering practice for how to do things. But so Docker on the laptop and then repository, and then check it out and build it to Singularity for a big machine. We use standard orchestration tools like Slurm for batch job, Kubernetes for services, and VM related workflows, Jupyter Lab if you want to run notebooks, Python notebooks or other interactive things. We have all the orchestration tools around that. We use some pretty not exotic storage formats. We do have object stores if you really like them with an S3 interface, but Google Buckets got mentioned earlier and having to port between these various things. We'll just give you files so that when you point to the name of a file, you just open that file if you give it the full path. So, and standard connectors for relational databases. So you don't have to go through this, check out of an object store, get it in the right place to run your workflow with us. But like I said, we have object stores if that's your thing. Codes like them more than people. So we have standard ways to get things in and out and to auth. If you're using multi-factor authentication, we can run through Google Authenticator or the Microsoft Authenticator if you prefer. SSH, SFTP, Grid FTP to get in and out. We run that TAPIS API I mentioned in the one example for web service access, RESTful access. We've also run instances of TAPIS in AWS and Azure, as well as TACC, and you can use that API for free. In fact, there's nothing we use that is a TACC-specific access workflow or API tool. So, if you can run it at TACC, you can run it anywhere else. We'll host the occasional proprietary thing for a user, but nothing that we centrally support is in any way TACC specific. So maybe we should be less like the cloud, and the cloud might want to look a little more like us in that regard. So we have computers, networks, storage systems, a really good supported Linux image that we know is stable, and you can run any layer of your choice on that. But we do have some recommendations, which, you know, as you might imagine, is the more you build on vendor specific services, the less portable you're going to be. And almost all of these have open equivalents, right? So I would look real hard at a technology that is not tied to one of the particular vendors or a particular site before you go down that path, so that you can move around. Try and use containers that run anywhere. Try and use methods that fetch those containers from central repositories rather than relying on something site specific. But even if you build your workflow to be portable, keep in mind that data migration has a cost in both money and time, and it adds up fast when your data gets very big. When you move from sort of your experiments to production, it can be huge. So think about where your data is or where your data should be. And at this point, plenty of our staff move back and forth from the cloud. Absolutely. So I think I have used all my minutes. So I just want to thank all my team that helps puts all this stuff together, because they make this all work. And I will stop there and take any questions.
Thank you, Dan, for that exciting and inspiring talk. Are there any questions for Dan?
Yeah, Dan, thanks very much for that really nice presentation. You know, a lot of the NCPI platforms, in terms of the data that's stored, almost a huge fraction of it is protected data sets. And it sounded like you did have some protected data sets. I wonder if you could talk a little bit more about some of the technologies and safeguards you have in place for that.
Yeah, I absolutely will. I'm answering one of the questions in the chat while we're at it. So we have about 125 protected data sets now at TACC. And if you actually log into the systems, if you see anything running through what's called the Carroll extra queue, you know there's protected data going on there. And so we can deal with HIPAA, FERPA, FISMA up to moderate. You know, in the NIST 871, 800-171, there's 17 categories and 63 subcategories, I think, of controlled unclassified information, and we do most of them at this point. We don't do classified research, but that doesn't matter for this kind of thing. So we have an intake form where we decide who has responsibilities for things like de-identifying, who is the owner, who's allowed to tell us who can access this and not. We've set up an encrypted file system that only gets attached to compute nodes when authorized users have access to those nodes through the queues. So that data is there on the compute nodes when protected workflows run, and then it just disappears and isn't, you know, you could root that node and you'll never find that file system if it's not your turn and an authorized user isn't on there. So hence the special queues to run those. But I've decided we're going to have some of those requirements on virtually every federally funded piece of work at some point. So we're trying to make protected data look as much like unprotected data as we can, because eventually I think it'll all be protected.
And as a follow-up, I mean, I think you said you had something like 350 petabytes currently.
Yeah.
If I think I know what the answer is, but if we wanted to talk to you about hosting a few tens more petabytes, sounds like you'd be open to that conversation.
Sure, and that big number is of course an aggregate and includes a lot of tape archive and things like that. But I think we have close to 40 petabytes in the sort of online front-facing stuff with Carroll. That's the publicly accessible collections, including 4 or 5 terabytes or petabytes of Galaxy storage, I think. So yeah, among that 40, and that's certainly something we can scale out. So we got to start saying exabytes. So I want to be among the first. Zettabytes are still scary, but exabytes we can handle. So
excellent, love it.
Let's move on to our next talk, which is titled FHIR for Genomics, the Path Forward. This will be presented to us by Mullai Murugan from the Baylor College of Medicine.
Good afternoon, everyone. I am Mullai with the Human Genome Sequencing Center at the Baylor College of Medicine. I'll take a few minutes today to talk about FHIR for Genomics, some of the initiatives that are being worked on currently, and the path forward. Let's first start with a general overview of FHIR for Genomics. But before that, a really quick intro. You're all likely familiar with HL7, Health Level 7 as the healthcare interoperability platform. And I'm sure you're also likely familiar with FHIR. However, I am not sure how many of you are familiar with
or have used the Genomics reporting IG. Genomics reporting implementation guide. Let's call it IG for short. At the high level, the Genomics reporting IG is a HL7 FHIR IG for clinical genomics reporting. It is a living work in progress IG that is being worked on by the HL7 Clinical Genomics Working Group. This IG is based on the FHIR code, that's what we have here, with specific profiles for genomics created from existing FHIR resources. And with these genomics profiles, this IG can be used to represent a typical genetic report. So here is a general overview of what the GRIG website looks like with some text and logical models. The intention here is not to scare you off, but to give you a basic idea of what is covered in this IG. As you can see, or in my case, maybe not see, this IG covers general genomic reporting. That is the resources and profiles necessary to represent a genetic report, starting with the patient the report is for, the specimen for whom you're taking the test, the service request, recommendations from the OP, and the results themselves. That is the overall interpretation, variants, and phenotype associations. Now, the genomics reporting IG is like a separate entity that is built on top of the HL7 FHIR specification. This IG goes through a completely independent standards processing cycle and creates specific profiles for genomics, which is kind of what you see here. And these are based on FHIR core resources. So in addition to genetics profiles, this IG also includes extensions. This is a way for you to add elements and fields that are not natively available in FHIR core. And it also uses coding systems such as LawInc, HGVS, etc. It also includes narrative guidance to satisfy typical genomics reporting use cases, kind of what you see here, variant reporting, genomic reporting, PGX reporting. However, this IG is still in trial use. As of May this year, the Clinical Genomics Working Group published release 2 of the standard for trial use. With the HL7 standards process, this means that the IG needs to go through several releases to pass through trial use before a normative ballot. That is a release that can be used in full-fledged production. So still ways to go. Before we move on from this topic, here are some resources for implementers who are interested in pursuing HL7 FHIR for genomics. The first link, along with the QR code, will give you a full list of all the information you would need to get started with implementing this IG or with participating in its development, which I highly recommend. Next, I would like to touch upon some new effort that is going on. As I mentioned earlier, the Clinical Genomics Working Group published the second version of its trial use in May this year. This release includes updates to the IG documentation, updates to general clinical genomic reporting, PGX reporting, and multiple ways of expressing information about the variant and more. Post-STU, this group has been expending quite a bit of time on the model of a genomic study. I'll get into this into a bit more detail in the next slide. But before that, a quick reference to a couple of other efforts. The first here is Genome-X. This is housed under the Codex FHIR Accelerator. Typically, the purpose of an accelerator program is to rapidly create and foster adoption of FHIR implementation guides. The goal of Genome-X in particular is to leverage HL7 FHIR, the genomics reporting IG, and to execute scalable pilots for a range of genetic use cases across multiple domains and including groups such as diagnostic labs, EHRs, etc. So if you're interested in participating or knowing more, there is a link included here. Next item, I'm sure you're all familiar with FHIR to OMOP. This approach is very helpful to get data from EHR systems to the OMOP CDM. May Terry with the MITRE group and others are working on transforming genomic data from the M-code FHIR IG to OMOP. Okay, so now a bit more about genomic study. The work on genomic study is led by Bob Freemuth, one of the co-chairs of the Clinical Genomics Working Group. And this new resource is targeted for the upcoming FHIR release sometime first quarter of next year. The main goal of this resource is to ensure that all relevant metadata related to a genomic study are aggregated together in a logical and structured way, and primary use cases, I have listed some use cases here, are factored in. Some use cases that start this development actually stem from consortia efforts like eMERGE3, where with multiple genome centers and providers collaborating, it becomes fairly critical to include details about the testing methodology, the analysis that was followed in structured result form. Or another use case, nowadays, a lot of genetic reports include multiple result components. That is, once again, using eMERGE3 as an example, the clinical reports we generated for eMERGE3 would include gene panel results and PGX results in the same report. So in such cases, if you want to clearly distinguish between the analysis methodology that you adopted for every result component, genomic study gives you a way to do so. So to accommodate these use cases and others, the Clinical Genomics Working Group started work on the genomic study resource. The goal here, once again, is to make sure that all metadata about a study is structured. That is what you see in the blue box, genomic study. And also that the study includes details about the analysis and methodology you followed, once again, a structured form. And finally, there are also provisions here to attach data files to this resource as well. And now on to the last item on my agenda. What are some of the challenges we face with establishing FHIR as an interoperability standard for genomics? When it comes to interoperability for healthcare data, for most of us, there is no doubt that FHIR has evolved as a standard since its inception in 2012, but it is still quite new. The first normative that is full-fledged production version, the R4 release, was only actually in 2019. Just would like to show something here. To get to normative, the FHIR management group established a set of levels, FHIR maturity model, FMM, which is what you see here. This progressively measures technical advancement known as maturity. We are talking about healthcare data here. Though such stringency is absolutely essential, as you can see from this figure, going from FMM0 to FMM5 is quite challenging. And FMM5 here is full-scale production with the normative release being used in at least 5 production systems in 2 countries. Now, when you think about genomics in this context, though there are more than a few challenges to get to a normative release, it becomes really critical for us to focus and grow the space, particularly considering that genomic medicine is now gaining more and more traction with a rapid increase in genetic testing. I pulled up the GTR. There are about 74,000 tests in the GTR now, and even more rapid increase in direct-to-consumer testing. According to a Consumer Reports survey, as of October 2020, at least 1 in 5 Americans had taken a DTC genetic test, and also increasing awareness with patients, providers, generally consumers. So establishing the standard and structuring genetic data is almost a prerequisite and foundational first step before we even begin to think about clinical decision support, apps, smartphone integration, data sharing, etc. So there are quite a few challenges here, but in my opinion, they are not insurmountable. Let's take a few moments to talk about these challenges and the path forward. Starting with the most basic challenge. In my opinion, there is quite a learning curve with understanding the clinical genomics reporting IG. As you can see from this figure, understanding the entities that would be required for putting together a genetic report and how these entities map to one another requires time and effort, particularly for something like defining the variant. So the takeaway here is that until this IG is normalized and battle tested in production, I would recommend that new implementers take a look at the IG documentation for learning and collaboration. The Clinical Genomics Working Group has spent quite a bit of time working to make this resource as detailed and as useful as possible for implementers and informaticians. And any feedback on the documentation is absolutely welcome. The next challenge is ease of implementation. Though the concept of FHIR is based on foundational web services technologies, almost with the sole intention of making it easy to implement, but based on a Genomics FHIR pilot, which we did as part of eMERGE Phase 3. This was a collaborative effort with Broad, Northwestern, Johns Hopkins, and Baylor. We did find the experience to be somewhat daunting. Understanding the ID specification, mapping the specification to eMERGE reporting requirements, and harmonizing both, it did require considerable effort. However, it should be mentioned here that the eMERGE effort happened even before the Clinical Genomics Working Group published its first trial use release. The spec is a lot more stable and robust now, I think thanks in part to the eMERGE use case that raised a number of questions that ultimately did lead to the robustness of the IG. So the takeaway here is that efforts such as eMERGE or even huge NIH, NHGRI efforts are absolutely essential to grow the spec, to identify use cases, to battle test it, and eventually take it towards a normative release, that is a full-fledged production release. A key factor to making progress on these standards are multiple pilot efforts. Pilot efforts are like the best way to test the spec and to grow it to maturity level 5. Once again, a big example of this is the eMERGE pilot. We were able to bring up a number of questions, gaps. Yeah, I just wanted to show that. So these were some of the questions that were raised as part of the eMERGE pilot effort. And these questions and these, the responses ultimately did get integrated into the ID itself, and it did lead to the growth of the spec. And to test validity for something like a genomic study, we definitely need multiple pilots testing the spec. The 5 examples that I have listed here for pilots are kind of outdated at this point, but the Genomics effort by Codex is recent and is a great way to accelerate the adoption of FHIR for genomics, as it seeks to work with the Clinical Genomics Working Group, and it seeks to engage multiple entities such as EHRs, diagnostic labs, to accelerate the adoption of FHIR. And lastly, though not exactly a pilot, efforts like connectathons are incredibly helpful with testing the spec. All the effort with developing the IG, the standards, pilots, it only goes so far without real adoption. This is a very interlinked ecosystem. And as you can imagine, true success can only be achieved if all parties are motivated. We need the EHRs and the diagnostic labs to engage with adopting the standard, or have third parties fill the gap with V2 to FHIR conversions for genomics and to make marketplace smart apps. However, on the upside, trend-setting research efforts, I've been hearing quite a bit this morning, using FHIR are on the rise. ONC's interoperability ruling as part of the 21st Century Cures Act, multiple research efforts, accelerators like Genomics, and there is actually participation of a certain EHR vendor with Genomics now. OMOP to FHIR for genomics, OMOP to FHIR for genomics by organizations such as MITRE. These are all great efforts to move this forward. And with that, I would like to take a moment to acknowledge the multitudes of people working on the standard, some of whom are listed here. I am merely a spokesperson today. Thank you for taking the time to listen to me.
Thank you for that very nice talk, Mullai. Are there any questions?
Thank you.
If there are no questions, then I believe it is time to move on to our next talk, which will be delivered by Heidi Rehm from the Broad Institute and is titled Supporting Genomic Data Sharing Through the Global Alliance of Genomics and Health.
Great. Thank you. It's a great pleasure to join you all today and just talk a little bit about the Global Alliance. I assume actually a number of you probably have been involved in the Global Alliance, but some of you maybe not. So I'll start with just a high-level overview and then give a couple of examples of work in my own space working with the Global Alliance and the benefits, and then hopefully leave 5 minutes for discussion so I can try to answer any questions. And I think there are a couple other people. I think I saw Peter Goodhand on this call, who's our CEO of Global Alliance. So anyway, as most of you may know, the Global Alliance is really focused on developing standards and framing policies for responsible genomic and health-related data sharing. And we do that by several critical ways: convening stakeholders to work together, creating standards and harmonized approaches through community consensus, catalyzing sharing of data. But we don't generate data or build the primary infrastructure or perform any research or clinical care. We really focus on just building the standards, though sometimes reference implementations end up needing to be built to help implement, disseminate. Next slide. So this gives an overview of the organizational structure of Global Alliance. We are now incorporated as a nonprofit. That means we have a board of directors, an executive committee. At the bottom, you'll see those of us on the executive committee. That includes Peter, who I think is on the call, who's our CEO. Ewan Burney's the chair of the steering committee. Then Catherine North and myself as vice chairs, Angela Page as our director of strategy and engagement, and Susan Fairley, who has joined us as our chief standards officer. There's also a very large secretariat, and the success of the Global Alliance is really in part because a lot of full-time, compensated staff who can devote their time to making sure this whole operation runs. So I've just sort of put a little picture of all those people that are so critical to this operation. On the left side, there is the strategic advisory and funding roles of the Global Alliance, which are obviously critical. We started out with 4 host institutions at OICR, Wellcome, Sanger, Broad, and EBI. We now have more stable, longer-term funding from NIH and Wellcome and AHR, MRC, and Genome Canada and CIHR. And that allows us to do a lot more. On the right side of this slide is the technical governance. We have a steering committee that's made up of the co-chairs of our work streams that I'll talk about in a second, as well as leads of the driver projects I'll also talk about. But a huge amount of the work is done by what are considered assigned experts and active contributors from the community who are participating in all of our work streams in the development of standards. And we have many of those. Next slide. So on the next slide, it really focuses on, and all of you working in this cloud-based space, I'm sure are very familiar, but because Global Alliance is global, we've really had to take a specific focus on the federation side of data sharing. And that's not to say that every approach to data sharing needs to be federated, but in many cases, if you're talking about individual level data, it often can't leave a country. And so we have had an increased focus on approaches that support federated analyses. But also, there are many individual cloud-based platforms and a lot of work in helping support work done in the cloud. But there are certain cases on the left side where centralized database is still the preferred mechanism, particularly for knowledge bases that don't deal with individual level data. Next slide. So this gives a view of how we actually work in terms of developing standards. So on the left side are our 6 technical work streams: discovery, large-scale genomics, DURI, cloud, genomic knowledge standards, and ClinPheno. We also have foundational work streams, our regulatory and ethics and data security. And the work of these work streams is really informed by the driver projects that we've designated. These are large-scale funded programs in genomics and health in different areas: rare disease, complex traits, cancer, basic biology, that are working closely with our work streams to both define where standards are needed, contribute to the development of those standards, and then pressure test them and implement them in their own projects, and then iterate when changes are needed. I also put at the very bottom left, we have a federated analysis systems project that's really piloting actual use of federated platforms and cloud-based analyses, as well as defining a starter kit. What are all the standards from Global Alliance that if you are getting into this space, you really want to launch and use? We have a task committee that helps harmonize across our own work streams. And of course, we have an EDI advisory board, given the importance of working globally with the entire community. Next slide. So this is just a look at our 2020 to 2022 strategic roadmap. Everything in green has been approved and is now in use, ongoing or in development. We continue to update these roadmaps with more work that needs to be done. It never ends, but this gives you a sense of all the different standards that are being approved and launched through the Global Alliance to support different aspects of genomic and health data sharing. Next slide. So I wanted to give a couple of use cases from my own work that have been critical in terms of the role of Global Alliance. So I work in the rare disease gene discovery space, and those of you who work in this space can appreciate that we haven't yet solved all the causes of rare disease. Most cases, unfortunately, remain unsolved, and we anticipate at least 10,000 more genes to be discovered. So, and the challenge right now is that most of these undiscovered rare disease genes are actually very, very rare, and most times a single investigator cannot amass enough data to implicate a gene in disease. So a number of years ago, actually 7 years ago, we launched, next slide please, the Matchmaker Exchange, and we did that really to support gene matching. The idea is you sequence, let's say, a trio with an individual with rare disease, you identify maybe a de novo mutation in a gene that's never been implicated in disease. Well, it's a candidate, but there's not enough evidence with just one case. But if we could find another case on the other side of the planet that has the same candidate gene and overlapping phenotype, you start to build the evidence to implicate a gene. But how do you find that other case that's on the other side of the planet? Well, if you skip to the next slide, that is where this federated Matchmaker Exchange comes in. And we worked with the work streams of the Global Alliance to define the API, to develop this federated platform so we could exchange data. That include, discovery work stream, or the equivalent of that at the time, our ClinPhenotype work stream, and also the regulatory and ethics in terms of the consent that was required or not to enable this platform. All of that led to the launch of the Matchmaker Exchange and a special issue of Human Mutations 7 years ago, outlining this whole platform. Skip ahead 7 years later to the next slide. We actually just this month published another special issue with, I think, 14 papers on this same platform, now showing the immense success of this platform. This led to the discovery of over 1,000 genes through this Matchmaker Exchange, really highlighting just how useful this endeavor was and how useful working with the Global Alliance was in helping launch this platform. So next slide. So now, this concept that we launched and have been supporting for the last 7 years is gene matching, which we consider a two-sided matchmaking. So two individuals or researchers or clinicians have candidates and you bring them together. But the next phase of this that we're now starting to work on, again, with the Global Alliance, is one-sided matching. I have a candidate, but no one else has identified it as a candidate in any other genome, but gee, if I could query everybody else's genomes, I might be able to find a variant in a gene in a case with matching phenotype that is actually a candidate, even though the person who had that data didn't recognize it. That's one-sided matching. And then even zero-sided matching, nobody's recognized the candidates, but if we can bring all this data together and look for commonalities, we can identify candidates by burden and analysis. Next slide, please. So we're working on this one-sided matching where you can go into a data set and query it. And there's been some isolated databases that have launched with their own interface. One is Variant Matcher at Hopkins. And this is just an example of me quickly registering myself as a user, entering at the top right. You'll see I entered 4 phenotypes in my patient and also a variant. And what comes back is a list of 8 cases with that variant and the phenotypes of each patient. Extraordinarily useful for me as a rare disease, both laboratory geneticist and researcher. And I was actually, because the phenotypes came back and 4 were healthy and 4 had random different phenotypes, quickly rule this out as a candidate. In other cases, you can rule a candidate in by getting good matches. Next slide. So we're really working to develop the next phase of a federated network where we can link up individual databases that have variant data and phenotypes and actually be able to query in this one-sided matching notion using standards developed by the Global Alliance, both the Beacon version 2 API, as well as another API called Data Connect version. And there's 2 papers in our special issue highlighting this new API, as well as early efforts to connect 4 different databases in the space. And you can imagine just how powerful this platform will become if we can hook up every biobank across the world. And I just showed an image of the IHCC and all of the biobanks of over 100,000 individuals around the world that are starting to work on data sharing and generating that data. So it's really just an example of where we hope to take this to really, further fuel rare disease discovery, and these same environments can contribute to other, not just rare disease, but other genomic discovery. Next slide. So I'll just end. I want to acknowledge an enormous number of people that are part of the Global Alliance. And we recently published our marker paper, which has many of these individuals highlighted there on the right. There are a number of other publications in that special issue of Cell Genomics, if you're interested in reading more about that. But for those of you who aren't already involved in Global Alliance, on the next slide is just a call to get involved. We welcome the work from all of you as you work on your own cloud-based platform here, and we'd love to hear what your needs are and work to support them and further engage with all of you. So I'm happy to stop there, and hopefully we have some time for questions and discussion with the group.
Thank you, Heidi, for that lovely talk. Very inspirational. I'd like to ask, are there any questions? Any comments for Heidi?
Yeah, I mean, J4GH has really, been enormously influential to NCPI, with the development of standards and resources and technologies. I'm wondering if there's a way that NCPI could also play a role in some of these, I forget we call them discovery projects.
Driver projects.
Driver projects. Is there a way that we could become more involved? That seems like a really meaningful way to advance science, and also kind of, it's the ultimate integration test to actually perform science using these technologies.
Yeah, we were just having, and Peter might be on to also weigh in on this, but we were just having a discussion. So already the National Cancer Data, forget what it's called, but is one of our driver projects, which I know is one of the NCPI cloud platforms. We were thinking that maybe, now several years later, it makes more sense to have the entire NCPI represent that driver project with Global Alliance, and we have been also thinking about another round of defining driver projects, because these have been the same for the last couple of years. So that is definitely a discussion to have with all of you, and thinking about the role that this organization plays within the Global Alliance more formally.
Well, speaking for Anvil, at least, I think we would very much welcome that discussion. I'm sure all the platforms in NCPI would as well. Thank you very much.
Great. Yeah, and Heidi's just, I see Heidi Stiffy is on as well, who is our program officer for the Global Alliance NIH grant, and Heidi, I'm sure will be helpful in that conversation as well.
If there are no additional questions, then I think it's time to move on to our final talk of our workshop. This talk will be given by Nick Weber from Strides, and his talk is titled Interoperability Opportunities and Challenges with the Cloud and Strides.
Wonderful. All right. All right, I think we're good. Well, thanks for having me. I'm Nick Weber. I lead the Strides initiative here at NIH, and I also lead our cloud services group within the Center for Information Technology. And I want to make sure this group understands that we have these two different parts of cloud, and I think many institutions will also have we'll need to grapple with some of the things that we're grappling with internally at NIH, and I want to have that be kind of the underpinning of some of these interoperability questions that we tend to hear in this community. So just in case anyone's unfamiliar, the Strides initiative is our set of partner relationships with commercial cloud providers, the three major infrastructure providers. We've negotiated cost discounts and set up training resources and other things as part of this program, and it's had some pretty good adoption over the last 3 years, but challenges remain nonetheless through Strides. And I'll describe again here the two core components that I think is kind of important. So Strides, for many of you, is kind of an agreement that you can leverage at your institution for your research program. So we have this agreement we've negotiated on behalf of NIH and NIH-funded research institutions. You can get to the cloud hopefully a little easier and cheaper, and that's kind of how Strides appears. We also have established kind of internally here at NIH these enterprise cloud platforms that we've had to go through the whole process of kind of authorizing, as many are familiar with the security and compliance requirements, and kind of setting up core infrastructure internally to be able to enable cloud as an organization. And I think that's one of the key things that I hope that we can mature Strides into as a way to kind of help research institutions who are grappling with the same things organizationally that NIH is grappling with to support our researchers to help get people to the cloud and establish common practices that I think will help to address some of these concerns about interoperability, working across clouds, working at interoperable levels, at infrastructure, at data, at application, and other levels that I know you all are kind of looking at with each of the data resources involved in NCPI. So these two things kind of inform one another, and then as part of the Strides program, we have professional services and vendor support and other things that I think collectively NIH and institutions represented here can really work with the cloud providers to be able to get some of the core products and solutions that are cloud native to support our work in a little bit better way, to support biomedical research work in a better way. So we can do some kind of collective bargaining and things at scale if we come together through this relationship with our cloud providers. And you heard yesterday from them on a panel as to some of the things that they're thinking about and ways that we might be able to leverage these relationships to follow back up. But one of the things that we talk about with Strides is there's a host of research programs, a host of institutions who have taken advantage of the program, but these are all still relatively siloed. And I applaud NCPI for Anvil and Biodata Catalyst and CRDC and the various programs that are part of this community. Similarly with programs like RAS, the Research Authentication Authorization Service that I'm sure many of you are very familiar with and leveraging. Those are ways to start to get this kind of piecemeal connections among these data resources. But I think as we heard from Titus yesterday, there's this question of the N+1 problem. How do we make it so that the next data resource, the next platform that stood up can be interoperable with the ones that were created previously? I think RAS has a large kind of contribution to that effort, but there's a host of other things that need to happen to be able to allow for interoperability across the cloud platforms. So I really just want to kind of describe a few things that I think are driving questions in interoperability, and then talk about ways that we might be able to start to overcome them as a community. And I'll couch that in one of the new offerings that we're putting out called the NIH Cloud Lab. So it's really a way for researchers to test out the cloud and explore kind of the native consoles, follow up on training they may have taken, and do some cost benchmarking and other analysis. And we've learned a lot in standing this up to really, I think, inform some of the interoperability challenges and try to break down some of those walls for working across cloud and working across systems in the various cloud platforms. So this cloud lab is really meant to be kind of a short-term, no cost to you. It's currently subsidized by ODSS, our Office of Data Science Strategy, to allow people to get access to the native cloud consoles, work in some growing and emerging repositories, GitHub repositories with tutorials of how to do some things within the cloud using kind of standard approaches and containers. So you can see at this URL some of the example repositories and modules for testing out things in the cloud. And we've begun to roll this out intramurally and also are starting beta testing for extramural research institutions on both AWS and GCP, again, to get a little bit more familiar with kind of some of the cloud native services and capabilities that people can do at the foundational level. And hopefully this will make sense here as I talk through some of the considerations and things we as a community might want to explore as we're seeing some of the challenges and drivers on the left and maybe some of the opportunities on the right. So I'll go from left to right, just kind of describing a few of the, again, the drivers and some considerations that I think this community might look at broadly, and we here at Strides would be very interested in helping to explore with everyone. So of course we have next year coming out the new data management and sharing policy, and that's driving a lot of, all right, how are we going to make data sets really more broadly available to the community. And I think, as you all know, we have some examples there with the domain-specific repositories, NCPI and other kind of core repositories that we're familiar with, as well as the generalist repositories, the FHIRs and the Dryads and other pieces. But one consideration that I would propose is, we've talked a little bit about these data meshes, and I'm interested in exploring, as we work with research institutions who are moving to the cloud, what does it look like to stand up kind of a data infrastructure and the capability to have data products, as we heard from the speaker yesterday, as nodes in this data mesh, to be able to scale that a little bit more broadly and establish some standards on how those are made available and inventoried and searched across, to be able to start to have a kind of distributed and federated network of data resources that, again, I think may come at levels similar to Anvil and Biodata Catalyst and the like, but may also come at more discrete levels of individual kind of research data projects or at lower levels. The second item, we have kind of concerns and challenges that we've always heard about. How do we reproduce data? How do we reuse things? How do we port from one cloud provider to the other if we're trying to conduct research and my data is here, but my compute is there. So from the Strides perspective, we've been really engaging with the cloud service providers to drive down barriers to data egress costs. Those are some of the big challenges that I know people are associated with, or people associate with kind of moving across clouds. And generally, we don't see data egress costs, data transfer costs for the vast majority of those participating in strides due to the egress waivers and due to other constructs that we collectively have worked through with the cloud providers to limit those costs in transferring data either via download or from one cloud to another. But I do also wonder, as we've looked at kind of core underlying technologies like OAuth and OIDC for RAS, what can we do to kind of have a similar program that NIH might support for standardizing ways that more than just this community, who I know is very familiar with Dockerization and containerization and orchestration like Kubernetes, but how does NIH support that being kind of another way that additional research programs can get the training and the support that they need to have a template to follow and work in this area. So I'm wondering whether some pilot programs and other things might be useful to break down some of the interoperability walls there. Additionally, for billing integration, some of the challenges that we've often heard is I have a cloud account here, and it ties back to this invoicing. I have credits over there, and it's just very complex to figure out how we can work across clouds when your billing and your kind of identity are separate. So we've kind of explored with the cloud providers ways to, and this is kind of tied to the Cloud Lab offering that I mentioned a few minutes ago, ways to allow researchers to authenticate using their home institution credentials, much like with RAS. So there's identity provider federation, and you're logging in natively to the commercial cloud service provider with that core identity that you're bringing with you. So it's not a local IAM identity on AWS or kind of a separate Azure Google identity. And I think one of the next steps that we can really look at to break down interoperability barriers there is kind of extending beyond that core federation and log into the cloud with your home institution credential, and kind of connect those on the Strides billing side to establish ways to kind of make sure there's one bill, there's one kind of pane of glass view of where your costs are coming from, so that doesn't add that additional complexity of moving across clouds or working across clouds in what you're doing. So that's one possibility there. Additionally, in Cloud Lab, for the cost enforcement barrier that we often hear, people kind of not wanting to overspend and really having a barrier to move to cloud in the first place. We've also kind of built with the cloud providers something that they've not traditionally been really interested in establishing natively on their platforms, which are kind of I guess ramp down Terraform scripts and other things within the Cloud Lab environment that we want to share a little bit more broadly, so that as you're getting to certain thresholds of expenditure, there are automatic kind of policies that are applied that say, well, you can't spin up additional resources because you're getting close to this level. And I think that's going to hopefully lower the barriers to people kind of being worried that cloud is going to cost way too much and we're going to overspend significantly. So again, we're eager to kind of share some of the approaches that we're taking to limit the cost in the Cloud Lab environment to a certain dollar threshold. In this case, it's $500 per account that people can kind of re-up, and that's kind of working functional code that we'd be interested in kind of seeing how people who are testing find that, and if that works well, how can that be leveraged elsewhere. And then lastly, I know again a driver toward not even moving to cloud in the first place, let alone working across clouds, is it's hard to know how much this is going to cost. And again, with the Cloud Lab approach, we're gathering data on, as people are running these various tutorials that we have for GWAS or variant calling or other kind of examples, describing here's how much it costs on a unit price example, so that people can kind of come and look at a library of how much one unit of analysis might cost, and kind of use that as a baseline, as well as, of course, coming in and trying their own analyses in that cost-controlled environment so that they can, establish ways to estimate costs for their grant application, or otherwise identify ways that moving to the cloud may be able to be cost effective. So again, those are a host of considerations that, again, I'd love to explore with the relevant parts of this community as we're eagerly pushing those forward on the strides front. But I'll close just by kind of coming back to that point of establishing enterprise cloud capabilities here within NIH. And this is something I think we've learned the hard way that I suspect that maybe your institutions may also be grappling with. So we have established kind of an operations and security function for our enterprise cloud environments, AWS, GCP, and Azure, that really have to focus on the platform, the security, the governance, the cost control, and this is a function that we've had to mature in all of these different areas that generally I would think that most people don't consider part and parcel to the research endeavor. This is infrastructure, this is technology stuff that somebody else deals with. But I think we have learned again here internally at NIH the hard way that we have to establish this as a baseline and establish kind of these core cloud platforms that our researchers can use. And that makes it so that access to cloud, at least this is the goal, is another tool in the tool belt of anyone here at NIH, so they can expect to have access to consumables and reagents and other things in their labs. They can also expect to have kind of native access to cloud secure and cost enforced and other cloud infrastructure capabilities to be able to support their research work. And I think this is a core underpinning at institutions to start to even think about what cloud interoperability looks like. We can do it at software levels and at data platform levels, so that you can connect data between some of the core data resources that NIH is supporting and that are part of NCPI. But I think until we have kind of established ways of working on the cloud kind of natively and institutionally, and mature ways to do things like infrastructure as code and DevOps organizationally, cloud interoperability is really going to continue to be a major challenge, and something that I think can only be solved without this sort of approach at kind of, again, pairwise levels. So I'll close with again just those thoughts, and welcome any questions or contributions or critiques. But I just wanted to share again some of the ways that we've been thinking about how to work across clouds, how to establish standards in clouds, how to make cloud barriers lower, so that we can achieve some of what help research programs achieve the outcomes that you're aiming for. So I'll leave it there and see what questions may arise.
Thank you, Nick, for a very insightful talk. Are there any questions? I see that a couple of questions have popped up on the chat. Tiffany asks, will a user utilizing Cloud Lab take advantage of cloud services directly or via analysis platforms?
Yeah, so it's native cloud capabilities. So the tutorials, for example, will have here's how to spin up Jupyter Notebooks or RStudio or Vertex AI or any of those things. But the Cloud Lab is really meant to complement the Terras and the 7bridges and the Nexuses that are more of the research focused platforms that kind of abstract in good ways some of the foundational parts of cloud, the building blocks. And that has benefits, but it also has drawbacks in that you can't maybe get access to all the native building block services or latest AI and ML capabilities, because you have to wait until those platforms make them available within their offerings. So again, it's meant to be a complement to those existing systems.
Could you say a bit more about, I think you call it the cloud enforcement, where you kind of spool down services?
Yeah, so we have two kind of approaches that we've been working on internally here at NIH. So one is, this is through professional services engagements with the cloud provider partners, where we can really do collaborative, development with them, hands-on keyboard work with them, since they don't have, natively in their product sets, just ways to say, here, turn this off once you hit a given threshold.
Yeah.
So one way is through kind of custom Terraform scripting. Terraform is kind of a cloud kind of landscape builder and way that many people use to establish all the different components on the cloud for the environment. And so we've been able to kind of use that to say, once you hit a certain threshold, don't allow spinning up of additional resources, or terminate accounts, or whatever you might want to do. Obviously, the software platforms that have been around for a while have those capabilities built in as part of their value proposition, but at the cloud infrastructure layer, at the building block layer, those don't really exist. We're trying to combat that with, again, some scripts and capabilities that people could leverage. We also internally use a third-party product that has some of this capability, not plugging any certain product over another, but we found some products on the market that, again, have a more of a GUI sort of approach to say, when you hit this given threshold, enact these restrictions. And that's been promising to those who've been using that product as well here internally in our kind of enterprise environments that I talked about, because you can individual administrators at our different institutes and centers can set those policies differentially and say, well, this one's allowed to go over this account, it's going to be shut down as soon as it hits a certain threshold, because it's kind of throwaway R&D, or dev or something like that. So that's provided some value internally here as well.
For the first one that uses Terraform, does that require that the services are sort of booted up with Terraform, or could you use, I don't know, Kubernetes or something else to get things going?
Yeah, so Terraform is the core, kind of, because we work across clouds, the core technology that we use, but it kind of calls, it's meant to call the native cloud services. So whether that's CloudFormation on AWS or other pieces. So you can containerize that, but it would be kind of still leveraging open source code, Terraform code, to be able to do some of that freezing or regulating, slowing down of account activity.
Got it. Thanks very much.
I see some
questions. This is Ian. One question. I mean, Strides, as far as I understand it, also supports if an institute or center has a system that they run. And for example, many of these happen within NCI, where they just want to port that system to the cloud from an on-premise data center. There's a certain amount of support that the Strides program provides for that kind of activity as well. And from a user point of view, they're just using the same application. They wouldn't even know that they're using the cloud. And when we look at the challenges of migrating users to the cloud, that's a good thing to have up our, in our quiver. Could you comment about that at all?
Yeah. So that and that kind of speaks to that last slide I was showing with kind of the operations team of people here at NIH. And it's not a huge team. I think it's a set of skills that we've had to develop and kind of try to recruit and retain, but having kind of staff who can work to do a migration of an application or a system from on-prem to the cloud, I think you maybe often heard kind of lift and shift, and we're just going to change where it's currently residing and move it to the cloud versus kind of rehost or replatform or re-architect. Those are kind of consultancy services that we offer to NIH research programs and staff to be able to kind of understand how they could leverage cloud architectures a little bit better and move systems from on-prem into the cloud. And we've done that for a lot of kind of research activities here at NIH so far. And that's not limited to research. So you may know that NIH's major system, grant system, the ERA system, is also running on the cloud through Strides with our in our AWS environment. And we've been able to kind of figure out and work with that team to move the application where end customers or people who are putting in grant applications don't know and don't care where that system resides, but it just so happens to be on the cloud. And I think that's a parallel to kind of other systems that could move from on-prem to cloud, and again, not having the end user have to know or care where that system is running.
Thank you, Nick, for answering these questions. Allison, I see that you've posted another question into the chat, though unfortunately, due to time constraints, I think that we'll have to leave it here. Perhaps we can follow up with this one asynchronously or through the chat or some other method. With that being said, it is now time to move into our final session of the NCPI workshop. And this is our concurrent breakout sessions. We have 5 topics in 5 separate breakout rooms. And I noticed that earlier in today's workshop, there were some discussions about, towards the end of Nara's talk, about some detailed extra slides that we might dive into a little bit deeper during the breakouts. My suggestion might be that we might look at using topic 3 to dive into some of those. And that's, there are some considerations. All of you signed up for a breakout room when registering, and we're doing our best to keep you associated with that breakout room. If you want to come and have this discussion in topic 3, please feel free to back out of your breakout room and we can get you into the room that you want to be. So without any further ado, Jen Vesio, are you willing to push us off into our breakout rooms one more time?
Yep, sending you now.
Thank you, Jen. Okay, we're back. Are we ready to do this? Everyone feeling good?
Sure.
I hear yes, absolutely, positively, 2 thumbs up. Let's do it. So here we go. Tiffany.
Yep. Okay. So time went by very quickly. We had grand plans of ranking the barriers to bringing researchers to cloud computing. I should say I had grand plans for ranking them. But yeah, so what we did in the session was we talked about the barriers to bringing researchers to cloud computing. And we also tried to quickly get in some ideas for strategies for getting around those barriers and things that we could do. So I'll just read what the list that we came up with. Expensive. So Mike brought this up yesterday with the cloud vendors that academics can often view on-prem as free, but everything that is not free is expensive. So that is one barrier with folks, not seeing those costs the way they do on-prem in the cloud. The second thing we have is cost education, fear of overspend. So not understanding how much stuff costs in the new way of working, not only, believing that it's expensive, but still once they decide that maybe it's worth it, not knowing, exactly how much things will cost and how to optimize that. And then obviously being afraid that they're going to spend too much. The third thing we talked about is the learning curve for doing science. So there's a learning curve and time spent obviously preparing to use the cloud, maybe even bringing over, stuff that you already have working like pipelines and things over to the cloud. Brendan and Melissa talked a lot about that earlier today. So there's that barrier as well. Value proposition. So is the value of the cloud worth the time to learn? Why do I need to learn it right now? and start transitioning now. So that's an important barrier. Policy, so aligning data policy with technology. So if the policy is not there to support, people going to the cloud, then that could be a barrier. Which analysis platform is for you? So that's more of like okay, if you've decided, if you're kind of trying to figure out if cloud computing is for you, the options, do you just natively learn the cloud, or do you, select a particular analysis platform? That's a hard thing to figure out, and can be a barrier to getting started. So the strategies that I'll just point out that we talked about a lot were around policy. So educating policy people and program officers to be more cloud savvy and understanding the value proposition of the cloud and why it's important. And so one example that was brought up was picking an IC with knowledge of the cloud and transferring that knowledge over to maybe an IC that doesn't necessarily need to know that much about the cloud right now. And so, yes, policy people speaking, policy people that really understand the cloud and its value, and having them meet with other folks. Another thing we talked a lot about, was the which analysis platform to use. So like having all of these choices, what do you do? Create some sort of educational tool, like a map that shows what's out there, maybe help guide somebody why you would choose one thing over another thing, or where do you just get started. So that was something that was brought up as an idea. For value proposition, we didn't talk about this, but Ravinder left it in the chat, which was great. So if we can educate folks on the jump off point when working on the cloud can improve their ROI of time and money, a lot of the other barriers might become easier to address. So I'll leave the last two that are up there for cost, education, and learning curve were just some ideas that I threw in there. So we talked the other day about incentivizing learning with training awards. So since that came up yesterday, I figured I'd throw that in here for the learning curve for doing science, if there were some awards that could help with that. And then we heard from Nick about the cloud lab from Strides. So that definitely should help with fear of overspend and help educate folks on costs of using the cloud if they're able to see them through the cloud lab. So yeah, that's an idea. And that's it for me. Thanks, Stephen.
Wonderful, Tiffany. I appreciate it.
No problem.
Next up is topic 2, reproducibility and interoperability of batch and ad hoc analyses.
Thanks, Stephen. We had a really strong group and an interesting discussion around, we had this rather vague topic to start with, and we found a nice direction to bring it all together. And I think it actually brings together some things that we've heard throughout the meeting. So the kind of take-home message was, for this, we feel that provenance would be a higher priority than perfect reproducibility. Having the first step to that provenance would ideally cover what is the data, what is the analysis, what is the configuration, what is the environment. But the first step that came up was really, what about the data that was used? And I think Michael gave some good examples of maybe something that had the same accession code, but over time, more samples are added. So you're not necessarily sure when you're going to reproduce it, what subjects were in there. And the two sort of blocking steps there would be, would we have a metadata exchange that would allow for, I analyze dataset X. So being able to share the dataset level metadata, maybe aggregate stats, how many males are in there, how many BAM files are in there, down all the way to subject level metadata. And then an accessioning space where you would have the DOI, or you would know, one of the problems that got brought up in the discussion is, do you know if the writer is speaking Anvil or speaking Kits First or speaking CRDC? Like, what is the one way that we can refer to this data so that it's clear to everybody, okay, we're actually talking about this? And maybe that goes and points out to a list of DERS URLs, but we wanted something that was a little more human friendly, intuitive and understanding than a 32 hexadecimal hex characters coming through. Allison brought up the 2 types of data releases, and Ben built on this a little bit. You could imagine where could we have something like a daily data release when you're looking at something like pediatric cancer, and that one new patient that's there is something that you want to bring into your analysis right away. And that's a different goal than people who want to analyze across the entire data set. They're looking at a different question. They want to have the completely harmonized, uniformly QC'd. Maybe that's the quarterly or biannual release. But overall, we think provenance would help for multiple situations, be that a data retraction, a data error. If you wanted to submit your analysis to the FDA, for example, if there were bug fixes or tool improvements. I think that was 2 cases that Michael brought up that would be good examples, or Bruno, of we don't actually want to perfectly reproduce it because we have a better version of the tool and we have better things that are going on, and we want to know how the analysis was done, even if we don't get to exactly the same answer. And then, wrapping this all up, this would be an important component of analysis reproducibility, where you could get to something like a checkpoint and restart. We have that for the workflow, but we don't have that for the entire analysis that's happened. So we feel like a strong thing that could come out of this is better provenance and some push towards, can we agree about how do we describe the data that was used by the researcher? And if anybody in the group wants to jump in if I've misstated anything, but I tried to catch that all in the 5 minutes we had.
Thank you, Jack. That was a very nice summary. I appreciate it. Moving along to topic 3, what technologies and data types are missing across platforms?
And I realized I've been on mute the whole bloody time. Sorry. Can you see my screen?
We can.
Okay. So, all right. So here we have, we had a very good conversation discussing what data sets and tools are needed. And so we first thought about, we first need to understand what's the point of bringing data sets together as far as for these platforms. And so we thought the things we could kind of break this down was like, linking data sets. What types of data sets are we interested in linking? And I see I have something in the chat. Just give me a second. And so we thought that these could be linked by geographical information, phenotypic information, and yes, I'm going to throw that in there, such in terms of health data, because they're never all in one place. But then this also led us into a larger discussion as to the benefits of, let me see here. If I can go to my next slide. Things are all over the place. Yeah, it concerns about linkage of linkings of data sets. There are different types of linkages when it comes to data sets, and those need to be considered. There's also technical issues of representing and storing data sets and making sure you balance this consent versus the linkage of those data sets. And they were concerned about provenance as well that come from linking. So it wasn't a simple thing as far as just linking the data, it's also understanding more about how you can go about doing these linkages that are meaningful and useful for the researchers, and while also doing it in a way that protects the patient's consent and privacy, particularly privacy. When we also look at another data sets we thought about linking as far as genotypes, we also thought about phenotype data. We think phenotype, when we looked at trying to link phenotype data, we thought that we also need to understand that phenotypes are not necessarily standardized, and that when you're looking across them, so there really needs to be better efforts at understanding that when you're trying to link phenotype data, that you actually have them mapped well, or mapped well to be able to do a meaningful analysis with different phenotypes across different data sets. Phenotype assertion is not necessarily comparable to one another. I kind of highlight this. Do you need to understand what that variation is? And there's the strength of evidence for these phenotypes also vary too. And so these are things that need to be factored in when you're thinking about that. We also realized that we couldn't just serve just the basic research side, but we also wanted to do things that focus on the clinical research side too. So we also thought about types of clinical data that may be of interest. And so we looked at the idea of clinical notes and EHR data that may be useful also. Okay, but we look more in that. This isn't as simple as just bringing the data together. It's also understanding how to use this data. And we also discussed the idea that, is NLP, natural language processing, actually at a stage where extracting this data set could be done in a meaningful way from clinical notes. And we realized that in our discussions that there's a lot of value in getting the data from the clinical notes, but the problem is that the patients have information that's all in the notes all over the place, and that it's very difficult to have the NLP to be able to extract this data set in a way that's meaningful. There's work going on to do this, and there's been significant progress in doing it. But it's more about that we should understand that that this is still work that's ongoing and should be encouraged to continue encouragement and advancing. We also highlight that there's that the billing codes could be useful. They're not the definitive way of actually extracting meaningful information from the clinical notes and the EHR data. So I wanted to summarize that that's where we came out of from our discussions. And I'm sorry if I threw all this together very quickly, but I want to turn it over to any of my colleagues who feel there's something that I missed in what I just presented. Okay, either everybody's so embarrassed by what I said, they don't want to chime in, or I actually didn't do that bad of a job.
Sounds great, Ken. It was great.
Thank you, Ken. Up next is topic 4, diversifying genomic data science.
Okay, so I'm going to share my screen. Seeing it right? I guess, okay. So we have a very small group, but very focused. Oh, sorry. Are you seeing my presentation?
Yes.
Okay, sorry. So we have a very small group and we have very focused discussion. These are the names that who joined the discussion, and we have NIH, MITRE, like the consulting and the journal participating in this discussion. So Kim, I have to especially thank Kim and Matthew with a very short notice. They joined our discussion and gave us a presentation on diversity, equity, and inclusive. So we started with Kim's talk. I just featured one picture to show what are real means by inclusive and treating equally. For it's not that everybody has the same box, but means everybody has to be able to see what is behind the gate. So next, we discuss few points, key points on what means diversity in the NCPI cloud platforms. So first of all, Marcia raised a good question on what do we know how diverse the data are in our NCPI cloud platforms? Actually, we don't know much. So, and then Rabia, she pointed out that we can leverage our NCPI's cloud platform interoperability power to pull data together for those small underrepresented populations from her editors, publications point of view. Oftentimes the underrepresented population study, they publish with very small numbers of participation. So I feel this is similar with rare disease use cases, because rare disease is also suffering with a small numbers of population. So I think, yes, we could. And then Jay mentioned about that all of us has a very diverse data set, we should utilize that. And then Matthew pointed out that if we are putting data together, there is ethical issues. For example, re-identify a person because we are a very small population. That leads to a discussion of data security, privacy in the cloud system. And then Michelle pointed out that if we are going to include the minority underrepresented groups, we need to provide a safe and secure environment for them from the people standpoint to ask them to involve in the science and the research. So we also find out that actually in our NCPI activities, we did not, we are missing the diversity related activities. So I think we need to address that. And then both Kim and Marcia point out that there are a lot of congressional funding support or from all agencies funding to support for the diversity related search. And we could also include specific diversity language for the FOAs in the future NCPI funding mechanism if we are establish that, when we establish that. So the next step, we have a very nice small starting point to address this is to do a very small data diversity investigation to all the NCPI platform data sets, and we can certainly report back to the next NCPI workshop. So if you all who are interested in this investigation work, please let me know. And we still have a lot to do in this area. That's all. And please add Marcia, Kim, and anybody else from the group if I miss anything.
No, I think that was a great summary.
So I'll present very rapidly the last topic 5, flagship uses for interoperability. I'm feeling a little bit sheepish about the fact that I cannot get figures together in exactly 0 seconds. So I don't know how you folks did that, but it's beautiful. So here's my figureless update. There clearly wasn't enough time. There's a lot to dive into, a lot to discuss. But today, we heard quite a bit about small fish. And it was discussed and brought up that we need to be sure that we're enabling small scale projects to effectively use what's already been built. We also discussed that supporting the big fish and enabling large scale, organizations to have large scale projects are what are going to open up the doors and pave the path for future researchers to approach science differently, more efficiently, more effectively, and ask bigger or better questions. And I think Brian O'Connor really nicely put it together when he said, yeah, both big fish and small fish. We need to support both of these. NCPI's success will be when we're able to achieve both. So we also touched on new NIH data management sharing policies that will enable broader sharing of process data outcomes. And it was noted that this will make an important contribution to interoperability. However, it might also be challenging to harmonize given that the data has already been analyzed. We discussed a little bit about generalist repositories, maybe most effective for partially processed or open access data. The repositories do account for the long tail of data sharing. There's a lot of data out there that in that long tail. So, and questions were asked, how can researchers find data across 7 or 8 generalized data repositories? And how can we consistently share metrics across these repositories? Something that is a little bit challenging. So it was a short session. We don't have perfectly well-rounded flagship use cases yet, but we know, but we were able to come up with some ideas around some directions, some areas that we might consider to continue working on. Does anybody from the team or topic 5 breakout have something to add in case I missed it? If not, that's the end of our update.
Okay, thank you, Stephen. I just have a quick few closing comments here to share with the group. First of all, thank you to all the presenters, all the discussants, everyone on the call right now. There's been just a number of remarkable discussions and presentations. I've learned a tremendous amount over the last couple of days. I would especially like to call out and thank ODSS and especially Susan Grederick for all their leadership, for all their insights, all their support to make NCPI possible. I'd also like to thank all of the NCPI platforms for their full participation in this meeting and their participation in NCPI as a whole, right? The theme of this is all interoperability. This only works if we're all working together. And finally, I'd like to give sort of special thanks to my team from Anvil that helped organize and run all the logistics. From Hopkins, I'm enormously grateful to Stephen Mosher, Jen Bessio, Natalie Kusher, kind of working behind the scenes. We also had a lot of support from our partners, especially Anthony Philippakis, Rachel Liao, Ken Wiley, Chris Wellington, and Valentina De Francesco. There's been a number of really exciting advances presented at this meeting. And I think in a nutshell, we're at this inflection point. I think that was actually Susan's term, where in the early years, it was about building prototypes and kind of speccing out API designs. And now we're really starting to see some of those discoveries being created through the interoperability research and kind of being distributed to the world through the first scientific manuscripts being put out to these efforts. It's been really exciting to watch, across the NIH and even more broadly. We've heard a number of discussions about the worldwide adoption of these standards and these interoperable technologies. At the same time, I'm also excited about individual institutions. I heard yesterday, for the very first time, for example, that Vanderbilt is like going all in on cloud computing and want their data to be interoperable. I think that's a really, really exciting time. And then even at the level of individual researchers, during this meeting, as I walk around Hopkins and at other meetings, I have people come up to me, ask about Anvil, ask about cloud computing in generally, people that I didn't think this would all be on their radar. It really, the word is really starting to get out. We're really starting to cross that bridge where people are really, really excited about the potential for what we can do here. Now, it's really a delight to me to see this. I think we have a lot of the key technologies in place. There's obviously a lot more to be done, and there's a lot more we can bring into the system. But we have a really strong foundation. So that is just really exciting to me. There's a lot of hurdles yet to be addressed, but nevertheless, we're starting to see the science being carried forth, and we're starting to see some really exciting results. The impact for this work is enormous, right? If you think about, what are the goals of these projects, they're improving the understanding and treatments of disease, a better understanding of just the forces of evolution, really it's just really enormously impactful, the type of biomedical research that has been going on and presented over the last couple of days. A couple of logistical points. So all of the slides, recordings, and notes will be posted on the NCPI portal. Give us a few days to get organized, but hopefully in a week or two, that'll all be available for you. The next NCPI meeting, I think, will be held sometime later this fall. That will be kind of a more closed meeting for NCPI participants. But I do hope in the next, say, next spring or so, we'll have another open meeting. Perhaps one of these could even be in person. I would be delighted to meet all of you in person. I think that would really bring another dimension that I've just been missing. So it's just one final thought is just I thank you very much for all your participation. It's just been a really outstanding meeting. I've just learned a tremendous amount. I hope you've been really energized by this. I thank you, and I hope to see you all in person in the very near future. Thanks, everyone.